Hello, good afternoon, and welcome back to this webinar on cutting-edge technologies for 2030 culture collections. So this afternoon, in this afternoon session, we will speak about another also new technologies that spread among culture collection, and which is the MALDI-TOF MS. And our first speaker, Dr. Anneline Wimmer, who is a service manager at BCCM LMG Bacteria Culture Collection, and she's a MALDI-TOF MS specialist. She's also a postdoc at the Laboratory of Microbiology at the Ghent University, and she will present a talk about the use of MALDI-TOF MS at the BCCM LMG Culture Collection. So please, Anneline, the room is yours. The floor is yours, sorry.
The room, the floor, it's all the same. Okay, thank you very much, Jean-Luc, for that nice introduction, and also a good afternoon from me as well to you all. Enjoyed the talks so far these 2 past days, so I hope I can deliver some quality equal to the presentations that I already witnessed yesterday and today. So indeed, we are going to talk about MALDI-TOF MS, an identification tool that we use in our BCCM LMG culture collection as a quality tool for quality control and in culturomics projects, a word that we have heard already the past 2 days. So what is MALDI-TOF MS in brief, and what is actually the steps that we take for this technique in our laboratory? So we have a cultivation step. So you will see the schematic popping up in the several implementations that we have for MALDI-TOF MS in our lab. But we want to adjust certain parts in the schematic depending on the implementation where we use the tool for. So we do some cultivation steps in the beginning, depending on the implementation, and is it to a 3rd generation culture or a 2nd generation culture, depending on. The cultivation steps are quite important. Why do we do it often to a 3rd generation? To just make sure that the culture is well adapted to the cultivation conditions used. Afterwards, for 99.99% of the time, we go for a cell extraction procedure based on the Bruker cell extraction protocol, for which we use a formic acid acetonitrile extraction procedure. Often we spot our cell extracts in duplicates, so they have technical replicates, and we go to our MALDI-TOF MS systems. We have 2 systems in-house. We have the old system that we purchased in 2016, and we have also the novel system, the smart system, which has an advantage compared to the older system. That it has the major advantages is actually the triple laser frequency, so it's measuring much more faster. Ideal for projects, culturomics projects, or for identification database construction. Once we have our mass spectra generated, we have different software tools that we use for different implementations again. So we have the Bruker software that is accompanied with the systems. We have also the speed software. It's actually an algorithm that is created in-house to process in a very fast and high throughput manner thousands of mass spectra. And we have also the bionumerics software application that we have from Applied Maths, my apologies, from MIRRI. Okay, so what are the 4 major implementations of MALDI-TOF MS at BCCM LMG? First of all, we use it to develop our own proprietary MALDI-TOF MS identification database. And the second part, we use it as an identification tool. So next to the genotypic services that we provide to our customers, we also provide MALDI-TOF MS as an identification tool. In the 3rd option, we have MALDI-TOF MS in our quality control for LMG strains, to actually preserve the quality of before and after preservation. We use that tool for that as well. And then in the last option, we use it as a dereplication tool in our culturomics projects, not only in BCCM LMG, but also in our research lab at the Lab of Microbiology at the Ghent University. So first of all, the development of our proprietary identification database. So as you know, those 2 systems that we have are accompanied with a commercial Bruker Daltonic identification database. We use it in research use only, and it comprises more than 9,600 MSPs. As you can see, there is a huge diversity present there, so a lot of species and genera are covered here. And the MSPs are mostly generated from clinically relevant microorganisms. However, Bruker is doing a huge investment and also including strains that are accompanied with veterinary diseases, for instance, or food pathogens, food safety, and so on, microorganisms. But we still decided to have or create our own proprietary identification database. As we also heard yesterday, that is actually the way to go if you want to uplift that identification process using MALDI-TOF MS. So at this moment, well, we started in 2018. We have already a nice diversity present there, and we have more than 3,800 main spectra, MSPs as we call them, entered in that database. Our main focus is food and environmental and also marine-related bacteria, recently added, is our main focus. So we really look at our research projects that are currently running in the research lab, but also we look at our customers' demand. So if we have a lot of food industry coming to us for identification services, of course we are flexible and try to add as many as we can food-related bacteria to that database. It is a continuously development, so we have a continuous development, and we try to have a sufficient coverage of the intraspecies diversity. This is not always an easy case, and we try to use data that we've obtained in the past via, for instance, the ALLP project that was running in the past, and try to add as many different strains for certain species as possible to really cover the identification potential of our database. The MSPs that we generate are actually generated according to the Bruker guidelines. So we had training sessions with Bruker how to do this. As such, this data is also complementary with the Bruker database, which is quite nice. You can just add them together and then just use both for identification purposes. Now, how are these strains selected, or what is our rationale for that? So we want to meet, as I said before, the dynamic identification needs of our clients. So if, for instance, a novel customer will come to us for have a certain identification purposes, we can, for instance, adjust our database settings or add novel entries to the database to make sure that we can provide with good quality identification results. We have those ongoing research projects, as I mentioned before. For instance, environmental marine-related bacteria. We have an EMBRC project at the moment running with Professor Anne Willems. And in that one of those projects, we really want to expand our database for marine-related bacteria. And we've done enormous research also on Antarctica in recent years. So those strains that we obtained in those projects are also added to our database. We have, of course, our pool, our treasure of BCCM LMG reference strains, which are quite nice that we can just use to expand our database as well. And we have, in recent years, our well-characterized strains that we have isolated in our culturomics projects. Those strains are then whole genome sequenced, so we are very sure about their identity and also nice additions to our database. So how do we proceed? What is the procedure? According to Bruker, as I've mentioned before, so we cultivate until the third generation, meaning that we have well-adapted reference strains to the grow conditions used. And one strain, a single grow condition, will correspond to one main spectrum. I've learned from other labs that they combine different cultivation conditions to create a single main spectrum. But to my opinion, you lose data. You lose peaks, you lose some unicities that are linked also to the cultivation medium used. But okay, each has its own favors, of course. So what happens afterwards? We do that cell extraction procedure. Each cell extract is spotted 7 or 8 times, and we measure the plate 4 times. So we have 28 to 32 mass spectra per cell extract. And then we analyze our data in flex analysis. So really look at the spectra, try to remove aberrant spectra with different peak intensities, for instance, if it's too high or too low. Differences in peak shifts. So we really look at the data very clearly, very in depth to make sure that we have good quality data. In total, 24 of those mass spectra are then combined in a single main spectrum, a single MSP, which we can store, of course, in our own database, using a software package of Bruker as well. And then the key point in this all is evaluation. So you really need to look at your data afterwards, make sure that the identification is correct, that is also linked to other species, other MSPs created for a certain genus, for instance. Try to make sure that you have a cluster analysis afterwards. Look where that particular MSP is grouping, because you know MALDI-TOF MS is no representation of phylogenetically relationships. So you really need to just make sure that those particular species group in a certain group that is present there. So you cannot really look at closely related species or strains. So you need to make sure that that is the case if you have an MSP added to your database. What is also important here in the evaluation is also look at, for instance, the closely related species or species that generate similar mass spectra, to make sure and take that into account in your assessment of identification projects, that it could be an identification between 1 or 2 strains or species, my apologies, or more. It's like the matching hints that you get from Bruker identification reports as well. Yeah, next to the evaluation, it's also very important to store your metadata. And in that case, we use our own online web-based platform, our own MasterDB that was created in our research lab, where we can store the preservation batch used for that particular strain, who was the depositor, and so on. So really make sure that you contain all the metadata that you gather for each spectrum. Okay, a second approach is using MALDI-TOF MS as an identification tool. This is actually quite straightforward. We have, for instance, a couple of culture media that are culture agar plates that are provided by a customer. And what we do is recuperate. Normal standard procedure is make a recuperation of each of those cultures, because it is well known that transportation of certain cultures can have an influence on the final protein mass spectrum that is generated in MALDI-TOF MS. So we want freshly grown cultures to go further with the analysis that we need to do. So we have, again, that cell extraction procedure. We spot each extract in duplicate, and we generate 2 mass spectra per cell extract. We do an identification using our databases that we have available, so our own and the one from Bruker, and we do data analysis in the Compass Explorer software. You get also a lot of information from the Bruker reports. And those Bruker reports really state identification scores. They also state the species consistency. So it's really important to take all those characteristics and parameters into account. Another level is also if you have a numerous set of cultures that are received from a customer, we also give an opportunity to limit budgets, so identification budgets or price settings. If you just generate a mass spectrum and compare them all together, dereplicate in this case, and just pinpoint and identify the species, the strains or isolates that are represented for a certain group or cluster that we've obtained. Okay, in a third part, we have also the quality control of LNG reference strains. This is a very important tool for us to make sure that we have a good and solid identification. Well, not identification, but more a comparison of the mass spectra that we generate before and after preservation. So to make sure that first the strain that we received from the depositor is authentic, we use sequence analysis for sure. But then in other place, we want to make sure that the authenticity is kept during or after preservation. Okay, so what is going on then? We also do a cultivation until the 3rd generation of the original material and also from the batch material, so before and after preservation. Again, we go for 3rd generation young, fresh culture, and we prepare a cell extract and spot this also in duplicate on a target plate. We run that target plate 4 times, the same way as we do for identification database construction. And we've obtained, in this case, 8 mass spectra per cell extract, so per original material or per batch material. We do the same data analysis as we've done for identification databases, and we obtain, at the end, we should obtain 4 good quality profiles in order to proceed with the quality control. If this is not the case, for instance, for the original material, we go for recultivation or retry, a second attempt, or go directly to sequence analysis and leave it as such. The same goes for batch material, as you can find in the bottom arrow part. If there is no good quality data, we go for sequence analysis. If we do have good quality data, we go with original material. What we do is create an MSP, a main spectrum, with only 4 spectra. That's a side note that I must make. Maximum, minimum 4 spectra, my apologies, and store it into the BCCM LMG quality control databases. For the batch material, if we generate a mass spectra, we compare those mass spectra generated with the different identification databases that we have, and also with our quality control with our BCCM LMG QC database. The thing is with this part, we often get no hits if we just compare a certain mass spectra or the mass spectra with our identification databases, because most often it's the strains that are deposited are novel described species. So we have no hits, but this tool makes it possible to compare the mass spectra. So not really go for identification, but compare mass spectra before and after preservation. And we already found some difficulties in certain LMG strains or due to deposit, because there were, for instance, mixed cultures present at the beginning already, or we've seen that the identification is not a match, or we have some difficulties in preservation as well. And this tool makes it possible to really, on a very short notice of deposit, try to find out what went wrong and really go for it, find the fault, and just make sure that it's working on a very short time period. If you need to wait for sequence analysis, that takes a little bit longer. At the end, we have our MALDI-TOF MS decision tree. So we made the comparisons with each other, with the databases, and then we get the 2 parameters again, as you would have for an identification step or an identification process. So we use our log scores, the Bruker identification scores, and the species consistency scores, and we go through a decision tree. My apologies, the decision tree is in Dutch, but the main focus should go on, okay, both parameters are taken into account, and if there is something wrong, the MALDI-TOF MS responsible is contacted, as well as the curator, to find out what could have gone wrong. Of course, we have more good flows than we have bad flows, of course. Okay, and then as a final part, I hope that I still have some time left, is the MALDI-TOF MS as a dereplication tool in our current culturomics projects. So I would like to highlight it via schematic and then also show it with 2 very small scale case studies that we've performed quite recently. So the dereplication tool, it is, as we've learned already yesterday and today, it is it's an addition if you have gone for isolating numerous of bacteria or microorganisms from certain samples. So we see it in 2 different approaches. At first, you have the samples. So for instance, samples that you want to investigate, or industry that wants to really go for the diversity in a certain sample, or want to do a targeted search and, for instance, only get lactic acid bacteria from certain samples that they could potentially use in their process. So we have the samples, but you also have the culture collections itself. So a lot of industry is harboring their own culture collections or research labs, for instance, that they gathered during the past years. And they really want to find out who is now in there and how do these isolates group together. So in that case, and we have already some good projects already run with industry regarding the culture collection screening, where we had some culture collections, we just opened them, we recuperated them on different media and generated mass spectra to really find out what is now the diversity of that collection, and also improve if there was if there already was some identification data available, or just provide identification data. So 2 different approaches that we have coming together with a high throughput screening facility to really gather all those isolates together. We have the potential to go for via agar plate. So as you've seen here in the picture, just basic cultivation, but we can also go for deep well, broad cultivation, and use our semi-automatic robot, ViaFlow, to really, in a high throughput screening manner, process thousands of isolates at a certain time, and also do the cell extraction and the MALDI-TOF MS target plate spotting in that single run. So it's quite impressive. So we have the MALDI-TOF MS measurements. Again, we obtain 2 mass spectra, technical replicas again, per isolate. And then we have 2 different approaches that we combine to perform data analysis. So in the left part, you have bionumerics that we use from Applied Maths MIRRI, where we do a cluster analysis. So you have global similarity taken into account of all the mass spectra that you generated. In a second approach, you have that speed algorithm that was created in-house a couple of years ago, and it was published also in 2019 by one of my colleagues. It is also available online, public access in GitHub. So really, if you want to use or try to use that algorithm to process your mass spectra, be free to look it up and find that GitHub portal to get the scripting. But it's a different mechanic behind it is based on unicities. So you look at your mass spectra and you go for local and peak matching, local and global peak matching parts of your spectra. And if you have a unique pattern there, you add this, or you call this your reference spectrum. And if you have all those unique patterns chosen out from your bunch of mass spectra, you just compare all your other mass spectra to that particular reference spectra. So in the first case, you get your similarity matrix, but in this case, you just get a non-metric similarity a matrix where you have all your unicities present and all the other mass spectra that have the same unicity linked to that one. And you get also an overview of your operational isolation units, as we call them. So I would like to sum up the presentation with 2 very small scale studies. So it's not thousands of isolates, but it's just to show you the principle of how we are working at this moment, to find novelty in certain interesting sources. So in April 2021, not that long ago, before summer, a pear farmer decided to just dump his 4 tons of pears that were still laying around in a refrigerator in a railway bedding in Limburg, Belgium. And they were rotting away on the side. So we decided, okay, very interesting here. We are just going to search for those pears. We're going to take samples of the pears, the rotting pears. We also seen bite marks, potentially of badgers that are living around that dump site. A lot of pollinators as well were flying around. So it was a very nice site for diversity in this case. We isolated those rotten pears, those pieces. We did homogenization and then also a enrichment in a fructose yeast broth. We applied 5 different conditions to that particular broth and obtained in total 81 bacterial isolates. So we really focused only on bacteria. And in this case, we tried to limit our targets to lactic acid, acetic acid, and the fructophiles. We try to tackle those ones. This is actually a pre-study that we have done because at the after New Year, a thesis student will work here to really go for the broad scale culturomics on those pears and on those flowers. You see here the cluster analysis that we've obtained, in which we have in total those 81 isolates represented in the dendrogram, and we have in total 9 different representatives that we've chosen based on the cluster analysis via speed and bionumerics. We had obtained some lactic acid bacteria, also some acetic acid bacteria. It may be too small to see here, but the ones with the red asterisks are the ones that we could not identify using our MALDI-TOF MS database. Meaning potentially that there could be novel bacteria. I'm quite sure that they will be novel because we really, for those genera added here in the dendrogram, we do have the full, validly published species that are comprised in those genera present in our database. So potential novel species descriptions are coming up here. In a second case study, on the same day that we did the analysis of the pears or the isolation of the pears, we also found some blossoms ready to, it was a season indeed, We did the same, so we also did an enrichment in that using the same cultivation conditions, and we've obtained 105 isolates in this case, and I will go through the dendrogram again. And we had, again, a nice, well, for the limited part of the study, of course, we had already some nice members here that could have a potential in valorization as well for industry. So we are targeted and we are looking for those type of bacteria. And hopefully after New Year, we can have a huge study as well on this one. Okay, so that rests me with just thanking everybody from our lab, from the culture collection, as well as from the research lab. A photo sadly taken before pre-COVID, so I'm very happy, or I will looking forward to have a new group photo of us all very soon, I hope. Okay, thank you for your attention.
Thank you very much, Anneline, for this quite comprehensive presentation of the development of in the use of the MALDI-TOF MS at BCCM LMG. So, Marta, are there any questions in the question and answer section?
Yes, thank you, Jean-Luc. There is one question from Jean Swings, which is: do you have many anaerobes in your database and related to the gut microbiome, for example? Thank you.
Good question. Thank you very much for that one, Professor Swings. At this moment, we are not focusing on the strict anaerobics or the anaerobes as such that are related to the gut microbiota. But recently, a novel professor started in our laboratory, MIRRI Webinar, who is very pinpointed on that target and has her expertise in human microbiome research. Also a member of the Jeroen Raas's lab. So probably in the future, we will start with that as well. So it depends at the moment on research projects, but we are going there in time.
We don't.
There's another
Marta?
question.
Marta, we don't hear you.
We don't hear. Okay, sorry. Yes, there was another question. If we have the time. So one from Matteo Schwilk, who is asking: are the examples of notorious species with extremely high intraspecies variability of MALDI-TOF spectra?
Yes, they are there. You have species which, indeed, we see that the more we add it, the more different strains, of course, we add for a certain species, we get better identification hits. If we have unknowns that were, for instance, isolated from a different part or from other sources, we do find this in MALDI-TOF MS. There are species that have, where you need to cover the diversity much more. That's true. In lactic acid bacteria, you have some of those who are present there.
Thank you, Anneline. Another question from a last question?
Yes, there is a last question from Nelson Lima. Have you a cost-effective study for the CQ on culture collections, keeping in mind that the gold standard remained the molecular biology?
I'm not sure if I understand the question.
Well, I think yes. The question is except the molecular biology, which is the golden standard.
Yes.
Do you have a quality control procedure for in your process for your spectra, for your identification?
Well, the thing is, for identification, I don't think that's the part, because identification is just an asset in this case, because we do the MALDI-TOF MS that's used as a before and after comparing mass spectra as such. We still use 16S RNA gene sequencing to make sure that we have the authenticity, my apologies, control of the strains when they enter our lab. But I'm not sure the cost effectiveness is, I don't understand it, my apologies. Maybe I should ask Nelson in an email separately.
Okay.
Okay, that's also a good question of Emmanuel. The thing is, in variations to mass spectra between and before and after, we use the same cultivation conditions, so the variations are quite limited. We do see some variations and try to keep in mind that MALDI-TOF MS is still a protein-based tool, so the variations are there. But we try to limit variations using the same cultivation conditions, but there is not such a kind of variation that is making it impossible to do the exercise due to preservation in this case. I hope that is also an answer to this question.
I think that we should now move on and go back to the next presentation. And I thank you, Anne-Line. I'm very sorry if I'm with your name.
And,
okay. No, and so thank you very much. And so now we have to move to the next presentation, and I have to welcome Dr. Sylvain Brice, who is the actual director of the CRB-IP at Institut Pasteur in Paris, and he will present an example of a web-based tool for the Klebsiella identification that has been developed at Institut Pasteur based on MALDI-TOF mass spectrometry.
Thank you.
Please,
do you hear me well?
Sylvain, the floor is yours.
You hear me well, and you see the slide?
Yeah, fantastic. And we see also the presentation. Everything is fine.
Okay, great. Thanks a lot, and thanks for the opportunity to present this work that we have been doing. Most of the work has actually been done by a postdoc in the lab called Sébastien Bridel. Oops, I've lost one slide, I guess, but it doesn't matter. As was presented by the previous speaker, identification based on MALDI-TOF relies on databases. And the issue is obviously then whether these databases are updated. And in particular, when we are dealing with taxonomy groups where taxonomy evolves fast, this may be an issue, in particular in the routine labs, because it takes time for the databases to be updated, because it's a complex process for the industrials. And so another issue is that the commercial databases are focusing mostly on clinical strains. And so if you're interested in more non-pathogenic or environmental species, they are typically underrepresented in these databases. Also, the software is a proprietary software, and so that means the analytical principles and methods are not very clear. So we are interested in Klebsiella because it's a very important pathogen in terms of public health threat. It's, as you know, a multi-drug resistant pathogen. Some infections are nearly untreatable or truly untreatable, and especially those that are caused by strains producing carbaphenemases or ESBLs. And so what is famous about Klebsiella is that it's a hospital opportunistic pathogen and causing a high burden in these settings, but it is also a community pathogen, sometimes called a hypervirulent pathogen for some of these strains at least, causing, for example, liver abscess, and typically in people with no risk factors. So importantly also, Klebsiella is an ecological generalist. We find it approximately everywhere, and including in human carriage. But some taxa are much more rare than others in clinical infections. So in the last years, the taxonomy of Klebsiella as a genus has evolved by the description of novel species or subspecies, and also the split of previously existing species. In particular, in the previous years, I would say 10 years ago, there were only two main clinically important species, Klebsiella pneumoniae and Klebsiella oxytoca. And these have been split in a number of different species that you can see here. And some of these species are actually very closely related. For example, Greymontii, Pasteuri, and Michiganensis. Also, the genus Klebsiella had been kind of split into Klebsiella and Raoultella, but recently there was a proposal to reunify Raoultella into Klebsiella and to abandon the genus Raoultella. So Due to this rapid evolution in the taxonomy, the databases that are used typically in clinical labs are not updated and don't include this diversity of novel species. So we are interested in creating a custom MALDI-TOF identification system to incorporate these novel taxonomic updates, and also to cover non-clinical species, because we know that Bruker and bioMARRI may not be totally interested in covering these species sometimes. And also we wanted to provide some transparent identification principles based on MALDI-TOF. So the strategy we used is a marker-based approach. So it's a bit different from the classical Bruker or bioMARRI spectral analysis, which rely on basically the entire spectra. Here we wanted to define markers, defined based on genomic data and based on MALDI-TOF experimental data, so that we knew what we were doing in terms of why we would call this isolate species A or species B. And so to develop this approach, we used a number of strains, which were genome-based identified. And the design of the study was a multi-center study where we tested the identification capacity from more than one MALDI-TOF apparatus. So to develop this approach, we used R, the programming language R, which contains a number of predefined packages. We didn't develop these. These were previously defined, such as MALDI-Quant, for example, and also the ability from R to develop web applications quite easily using R Shiny. So in practice, we used a number of strains, which you can see here. This slide is probably a bit too detailed for this presentation here today. But basically, we had a first set of reference strains from which we defined markers that were specific for each of the species. And then we used a second data set of a bit less than 300 isolates from Australian humans and animals. So representing a very diverse and different set of isolates. And so the strategy we chose was to have a two-step strategy for identification of Klebsiella samples. The first step is to discriminate between Oxytoca and pneumoniae complexes. And the second step is, once we know the complex, use specific markers that discriminate within each of these complexes. So, for example, to distinguish Klebsiella pneumoniae from Klebsiella quasi-pneumoniae or varicola. So perhaps this slide is the most important of this presentation, and because it encapsulates the originality of our approach. And this approach, Sebastian Briedel has called it multi-peak shift typing, a bit analogous to multi-locus sequence typing, when we base the typing of strains based on genomic sequencing. So in this approach, we really identify specific proteins, peaks corresponding to specific proteins, and these can be double charged or single charged with a single star here. And so the ratio of mass to charge is quite different. For example, you have some values in the 8,000s and some values in the 4,000s. So each of the peaks that we use in the system is identified to a protein and to a number of charges. And so the point of this approach is analogous to MLST, is to give isoform numbers to the different protein forms. And these numbers are then combined into a profile, which is analogous to the sequence type, which we call this MPST. Right? And so once we have developed this approach, the game consists of cataloging the number of the different MPSTs that we find in each species. So typically we have only one or a few MPSTs per species, and so this is quite easy to handle. And we did the same for the Oxytoca group. I don't show the slide for that. Everything is in the publication. So to validate the approach, we used, as I said, a bit less than 300 isolates from Australia And regarding the Klebsiella pneumoniae complex, we found very good rates of correct identification. The reference identification in this case is based on genomic sequencing, so it's quite precise. And basically, 90% of the isolates were identified correctly at the phylogroup level, meaning at the species or subspecies level, and only 10% were not. Which I don't have the comparative figures here, but if you would use a Bruker or a bioMIRRI MultiMS system, you would have much lower rates of good identification at the species level. All right, so regarding the Oxytoca complex, the results were much less encouraging. We did not have a very good level of precise identification. This is in part due to the fact that we had very few strains to create the reference spectra, and we need to work more on that. And also we are now working on a machine learning based on full spectral data approach to see if it provides better results than the marker-based approach. So we have developed that into a website, which we called MALDI-TOF, or Klebsiella MALDI-TOF, but obviously this approach can be expanded to other organisms of interest, and this is obviously a call for other MIRRI partners. If they are interested to develop these things together, we would be interested to discuss that. So MALDI-TOF will consist of a web interface where you will find this type of screenshots, and I thought it would be good to make a demo. Before that, I will just mention some of the principles, which are quite obvious, in fact. So the first step is obviously to look for the spectral folders, which are in your Bruker. In this case, we use Bruker data only in your Bruker apparatus, or the computer attached to the Bruker apparatus, at least. And first step is we introduced a step to visualize spectra, just to make sure they are correct and good quality, and to get a feel of them if necessary. The system is quite fast. I have put here some metrics. So you require approximately 10 seconds once your spectra is uploaded to the website to get an identification result. And you can upload batches of up to 100 isolates without any problem. So let me try to make a demo. I'm hoping it will work, because the BB system is a bit complex. So normally you should... Do you see my screen here?
Yes, we do.
Yes, yes.
Okay, great. So the first step is to load your data. So you will fetch the data by browsing. In this case, I have 3 isolates that were extracted from my Bruker apparatus. And so here you have a kind of index ruler where you can visualize any of the spectra that were just uploaded. There were only 3 files, but 12 spectra, because some of the zip files contained technical replicates, and they are pooled into a single isolate in the system. And so here you can see a table with the status in terms of quality of the data. I think peak 1 is a bit of bad quality. You see a big background noise. And so this is a warning here. So the next step is this first step that I told you about, which consists of determining if it's an Oxytoca or pneumoniae complex member. And in this case, the 3 isolates were identified as Oxytoca, pneumoniae, and pneumoniae. And here you see there is a kind of metric score value to define the strengths of the reliability, let's say, of the approach. And so the next step is obviously identification at the phylogroup level or species level. In this case, this was now done. So here you have the results. So the 3 isolates were identified as pneumoniae, varicola, subspecies varicola, and the third one was not identified because it belongs to Oxytoca complex, I believe. And you can have a detailed table where you get all the peaks that were identified in your spectra, the MPST profile, and the result. And you can obviously download that as a CSV or tab delimited format, etc. Now, the next thing is you can also visualize the data. If you have, in particular, if you have atypical strains, it's quite interesting to look at what's happening with their spectra in comparison with reference strains. And so here you can see your isolate in black, typically at the bottom of the list, and you can compare the peaks with a number of reference strains that we have loaded into the system. And so it's easy to zoom in these regions or unzoom, etc., etc. So it's a very handy visualization tool that was incorporated into the system. And I believe we can go back to, yeah, you can unzoom or zoom again on the particular window or look at the entire spectrum. So just to finish, there is a FAQ, frequently asked questions page, where you can find complementary information and also credits. In fact, this system was based on a previous version that was developed at INRAE by Sébastien Bridel, Eric Duchot, and colleagues in Jouy-en-Josas. All right, I'm not sure I'm not late, so I will now try to go back to the presentation. So these are screenshots of the demo that I just did. Yeah, that's it. That is my last slide, just to mention that the work was published, and you can see the publication here. The reference is in Research in Microbiology. And this was a collaboration with the group of Kat Holt, with in particular Stephen Watts, and some other people in my group or in Kat Holt's group. And also the code base for this system is available. It's open source, and you can find it in the code base archive of Institut Pasteur. With that, I also thank the funder, One Health EGP, for this work. Thank you very much for your attention.
Thank you, Sylvain, for this nice presentation of this very nice tool. And I hope that people will have questions. But my immediate question was about how generic it is. I mean, because you have chosen specific markers. I mean, how far can we expand? Should we choose other markers for other genus, or could we look at the same orthologous markers in other genus and keep on expand the database? That means that you can, it's not be Klebsiella typer, but let's say a more wider tool for species identification.
Yes, thank you for the question. So this is completely generic, but you would have to define the markers for each group of bacteria that you are interested in. Typically, the proteins that we used here are not ribosomal proteins, but other highly expressed proteins that we found in the spectra, which were useful for discrimination because they are more variable than the ribosomal proteins. So I guess you need a kind of empirical approach. The first step is really to look at different isolates that represent the diversity within each of the species you are interested to discriminate. And then you would select from the inspection of the peaks of the spectra, those peaks that are specific and allow discrimination of the different types in combination. There is no limitation in the number of markers that you can introduce into the spectra, the profiles, right? I mean the MPST profiles. So you could have 10 or 15 markers if you like, instead of only 5 or 6 that we had in our system, for example.
Okay, thank you. Thank you, Sylvain, again. I see that time is running, and so I think that I invite people to ask questions in the question and answer section. And this question will be kept for the discussion at the end of the session on MALDI-TOF. And now we are going to also move to the presentation of Vincent Robert. So Vincent Robert heads the Westerditch Fungal Biodiversity Institute Bioinformatics Group. I'm sorry for the pronunciation of Westerditch. And he's also the CEO of BioWare, and the company that developed the Biomics software made to manage BRC's data. Today, we present a joint project that may lead to the development of shared tools and databases to identify microbes using MALDI-TOF. So please, Vincent, the floor is yours.
Thank you, Jean-Luc. You hear me?
Yes, we hear you.
I try to
we
share
just
my
wait for your presentation.
I know. I try to share my screen, but it seems to be frozen for whatever stupid reason.
Maybe
when I clicked on share, it's becoming blurry, and so I hope it will come, but
maybe did you? Here it is.
I'm not sure.
Well, it looked like a presentation with your name on the first, with your name on it.
Well, really, it's really just turning and looping. Well, maybe I will try to start without any slide, I suppose.
Excuse me?
Or maybe I should restart the presentation.
I shared your PDF presentation,
you know,
so you
the
just
system.
have to change the slide.
I will restart. I come back in a minute. I would say.
No. It's working.
Please.
You can just.
Okay.
Your presentation is shared on the screen. You just have like to use the arrows and like go over the slides.
I think he's gone.
Yeah. I will re-invite him.
That's a pity.
But you see the slides, right?
Yes.
Yeah, you can make the presentation for him.
It's disconnected.
It was running too smoothly.
I will try to call him.
I think you should reconnect immediately, but here. Where is Vincent? Hello, Vincent. You did wonderful effects. It was very nice. We saw your slides.
Okay. Now, do you see it again?
Yes.
Okay, I'm sorry about that, but you know, the system froze completely. So, well, now it's starting. So let's go straight. So I'm sharing this presentation with Stone Boekhout from the Westerdijk Institute and Carlotto Krauter from Brugge that helped me to create this presentation. I'm not a specialist, unlike the two previous speakers about MALDI-TOF, but I'm in this project because I'm a specialist of databases and software, and also used to be, a specialist of yeast in the past. So the title here is a bit optimistic towards a universal and community-based MALDI-TOF database and identification system. Universal it's probably too much, but that's the final goal, maybe. So the idea is that we would like to use different types of methods for identification. Of course, we can still use morphology, we can use chemistry, physiological, serological based systems. We could also use DNA barcoding or metabarcoding. I think this is still one of the major systems for identification. And in the future, it will be probably even more important for identification, I mean, metabarcoding. Metagenomics will also become very important in the future, especially when more genomes will become available. Of course, in bacteria, there's already a lot of genomes, but in fungi, for example, it's not yet the case. And then genomics approach and so on. But here we are talking about MALDI-TOF. I'm not going to explain what MALDI-TOF is because I've already spent too much time on those things. And anyway, you know what it is, otherwise you would not be there. the main point is that, Bruker alone in 2020 had already sold 4,000 Bruker biotypers, which is about 2/3 of the market. The rest is shared by bioMérieux and then a little company in China that is also producing MALDI-like systems. So Europe is the main market for Bruker. They have more than 1,500 systems there, and mainly people in the clinical labs are using Bruker or at least MALDI-TOF systems. So this is probably why this is more directed towards pathogens rather than environmental things. But I'm certain that the system could work as well for non-clinical isolates, of course. So you need to know that just in 2020, about 200 million identifications were performed with the Bruker system only, and which holds about 2/3 of the machines. So it's about 300 million for all the machines altogether just in 2020. So 300 million identification. It gives you an idea of the number of runs that are done using these systems. Most of the identifications are done at the bacterial level. So the databases are mainly well established for bacteria, not so well for yeast, and even less probably for fungi. So we have developed a number of MSPs for, we have a number of MSPs for yeast at the Westerdijk, probably more than 150,000 at the very moment. I've not checked recently, but it was the number 1 year ago. And so the good thing is that the specificity is above 98%. Of course, it depends on the labs, it depends on the quality, it depends on many of the things, but this is an FDA study that found those numbers. So one of the problems, if you want, is that the equipment costs 200,000 euro or so, maybe a bit less, maybe a bit more, I don't know, depending on the options and depending on who you are, I guess. But the good thing is that the cost of the per sample, for routine diagnostic, it's very low. It's only a few cents, so it's very cheap to run it. So the pros are that it's easy to use. The running costs are neglectable. It's now widely used for routine diagnostic. Results are obtained directly after running the system, and this is a high precision and accuracy system. There is no need for highly experimented operators. So you may have people that have very little experience, at least in terms of taxonomy or in terms of, although they need to have their critical brain open when they receive an identification, for sure, as usual. So the count about this system is that this is very expensive to buy the machine. You need a pure culture because, mixed culture could work to some extent, but not when you have more than 1 or 2 or 3 organisms involved. So it takes time to do the pure culture. And so it's not always easy in clinical settings to get that, and it takes time, and time is really important. So it's a delayed answer compared to immune tests or that are less reliable, and sometimes depends. And metagenomics is in its infancy stage. So I would say that metagenomics would be a great tool, or meta-barcoding at the very least, but this is still not possible to do it in routine diagnostic, I would say, or not very well done at this stage. So the challenges are that the reference database of Bruker is not listing all the species that are out there. So, and when we have an identification, we don't have much information. We know things like, okay, this is a Candida albicans, for example, this is CBS something, or things like that, not much more. So we need to enrich all those data when the identification is performed, and we need to dig into the information more deeply. So the idea is to increase the size of the database and to introduce more diversity in terms of strains and in terms of species as well, of course. So there is no mechanism in place at the moment to gather data from users and identifiers to potentially enrich the reference database using, for example, the geographic origin of the strains, the substrates on which they were isolated, and so on, the habitat, the patient, and so on. Of course, we are not interested in collecting personal data here, but, all those data are extremely important when you do some epidemiological studies. You want to know when they appear, where they appear, at which season, maybe in which year, and so on. And potentially give some treatments and so on, and detailed information. So also the profiles are kept as such, like this, but they are pre-analyzed or pre-processed, and the peaks are found, and this is the peaks that are really used for the identification. This is nice, but those profiles contain more information than peaks that are there just for the identification. Potentially, we could even use it for antibiotic resistance potential and things like that. Why not? So we have now a project with Brucker and the CDC in Atlanta to create a reference database, and of course the Westlake Institute, so formerly known as CBS, to create a system that would work together. Maybe MIRRI, people or collections or partners would be interested to join, and I'm pretty sure that a number of them will join later on. And why not? We could open it, and we would like to open it to other partners in the future for sure as well. So the basic idea of the system would be that, of course, what we would need to do is to take all the reference profiles from all the partners, do manual and automated checkings for the cleaning of and clean all those data, make sure that they are dereplicated, or at least that they are consistent and clean. So a big step of curation is very much needed, as was mentioned already by the two previous speakers. And then the databases would be available to any user on a website. And so an unknown profile would be able to be compared to 1, 2, 3, or many different reference databases at the same time. So the idea would be to compare unknown profiles, get a list of the best matching profiles coming from the different databases, and then you would get a matching thing, a bit along the lines of what Sylvain has shown before, and also Annelien somehow. The idea also is to keep track of the position where the identification, or at least where the sample has been collected, and also keep track of the seasonality of the samples, and also about the years where they were collected. But for that, we will need to gather the information from the end users of the system, that if they are willing to share their samples and their data, we would be collecting them. That would go to an end user database, and finally it would be checked for quality, and it would become available again for the other end users as well, if they want to include those in their system. So there is a pipeline behind that, and there are different steps. Some of them are easy to do, or more or less easy to do, because there are already some tools that are available. This is in orange. In red, we need to develop a number of things around that. And so there is an idea to develop a pipeline within the EOSC Live project, to which we are collaborating. And we are not starting from nowhere, because the CDC already has the MicrobeNet solution. which is allowing to, well, I'm sorry, one of the slide is has been lost. The yeast website and the yeast reference system contains more than 2,000 species of yeast. So in fact, all the species of yeast are there with plenty of data, physiological barcoding and other types of data. We also have the MIRRI system that contains a lot of data as well, and that could join in the same kind of way. We also have connections to reference websites and books, like the Atlas of Clinical Fungi, that would be connected to the species names, or Mycobank, where we can do pairwise sequence alignments, or other types of systems. Like another one that was not there, but I missed anyway, which is a polyphasic ID system where you can include physiology, DNA, and MALDI at the same time. So the current architecture is normally that when you have a MALDI-TOF, you do the measurement, you do the pre-processing, you get the peak list, and then you do the identification on the local server. At the CDC, it's very similar, but you can, in addition, upload your profiles, a bit like Sylvain has shown for the MALDI typer. And you can upload your profile, and then you get an identification using their reference database that contains a series of locally stored databases. So what we want to do is to do a pilot project together with Brücker and the CDC, and of course the Westerdijk, to create a reference database. So the first steps are the same. You do your identification on your local server or against your local server as well. So this is the client server. You can compare your profiles against the CDC one, against another one like the Westerdijk, or potentially like another one again like the MIRRI one. And those would be in a central server or potentially distributed server. So the data would be sent to those different servers or to a central server for identification. The identifications would be combined into a single list of identification, and hyperlinks would be redirecting the results to the original database, showing the original strains that have been used to do the identification, or to get the profiles at least. The problem here is the euro. It's not, totally free to do all those things, because, running all those identifications, when you do a few identifications, it's very easy and it's feasible. But if you think about the 200 million or the 300 million identifications performed every year, and this is growing, then it's becoming another problem. And there we need to see the business model. We have some discussions with Brücker, and they indicated that they might be willing to support us, but this is not granted yet, and we need to see if that could be the case in the future. So it's very interesting. So finally, if we want to do this, the good thing is that we get more precision with the quality of the MALDI system. The speed of identification is high, and this is very critical for patients to have a quick identification. This is low cost, provided that you have the money to buy the machine. It can allow you to get better treatments and therefore better survival rates. So with this, I would like to thank Brücker for sure, the CDC, the Westerdijk, of course, and the EOS Life project, the MIRRI project as well, and the yeast website and BioWare as well. So with that, I'm going to take your question if we have time.
Thank you, Vincent. Actually, well, I don't know if there are different specific questions to your presentation, but in any case, I don't know, Marta, if there are any specific questions for Vincent?
Yes, thank you very much. There is one from Cledier Santos, who is saying, excellent approach indeed. Congratulations for this, Vincent. And is this a database with open and free access like GenBank?
That would be my dream, yes. So the a priori is yes, it will be open and free. I don't know if there will be a login password, probably, maybe, but that would not be subject to money request. The model of the CDC is to be free as well. You just need to have a login password as well.
Thank you.
We can always, accept the possibility that some reference database would like to get some money from the system, and that maybe some databases would not be for free. But a priori, that's not the goal.
Thank you.
Martha?
Do we have more time, Jean-Luc? Yes. Okay.
But
So next.
Yes, I think we're going to take two questions, and then we'll go to the round table, let's say.
Thank you. So from Pierre Becker, does this initiative focus on yeasts only, or also bacteria, molds, etc.?
So at the very beginning, it will be on yeast because we want to look at the ways the system could work and to do a pilot project, a mock project. But the final idea is not to limit ourselves to yeast, no. But the yeast is the first one, yes.
Thank you. One more, Jean-Luc. Thanks. Mateusz Wilk, exactly. Any plans to link this project to GBIF?
Sorry, can you say it again? There was
Are there any plans to link this project to the GBIF?
The Global Biogeography Information Facility.
Well, we've not thought about that, to be honest. So maybe.
So I think now, because Vincent Sylvain asked one question, I think now it's just time to open the question answer for everyone, to wish to not too specific to Vincent, but actually this comes to because indeed, if I understood correctly, because I'm not a MALDI-TOF user, there is a need for a shared database and to expand that and to share, to have a because as many I've just tried once MALDI-TOF, and I only saw that my favorite environmental yeasts were not inside the database. So it's why we need to expand the database, and for that, sharing information is a possibility, and tools to that are would be fantastic approach to do that. And it's why the approach of Sylvain and of Vincent are so useful, I mean. So now I have the question. I would raise the question of Vincent, but he can't say it directly. And so I might have a personal question also. Would be then after you have answered yours, it would be, how is it possible to combine your approach of both systems if it's thinkable? So, but Vincent, the question of Vincent was, so Sylvain, sorry, was Vincent, very interesting initiative, but how would you guarantee quality of primary information?
Okay, so this is the responsibility first of the providers of the spectra to deliver quality data. That's the first line of defense, if you want. And then there would be a second line of defense, which would be to compare the data against the others, you see, because when you have several reference databases and sometimes they have the same pieces and things like that, if there is no matching, that makes no sense. I mean, or maybe that could make sense, but we would compare the profiles of the different providers and see if it's consistent or not, and if not, we would raise a point to the curators of the original provider. And it's only when everything seems to be consistent that the profiles would go into the final reference database.
Okay, maybe I can take thank you. I mean, yeah, I understand this. In GenBank, strains sequences are deposited by any people, and there is little curation on the or control on the identification, for example, taxonomic identification. For example, for some genes for which it's not easy to determine the identification. And also another issue is obviously when you define a new species or split a species into two different new names, how do you keep up with the updates of your reference spectra? And I mean, this needs a lot of curation time, I guess. So the question becomes, how do we guarantee sustainability of the system? A bit like GeneBank, for example, you know, it's federal funds and NIH funds, etc. But in this case, I mean, it would be very interesting to embark in such an initiative, but in some way, it's not worse if there is no sustainability. So perhaps Bruker can also play a role because they may have an interest in incorporating some spectra into their commercial databases. That can also be a mechanism. But
exactly, I think that my best bet is Bruker, because their interest is to be as useful as possible and to make their system really very strong. We were really impressed by their willingness to collaborate with us. You know, they were really opening things. We were really astonished by that. And so this is, of course, to their advantage, because if they provide a good database made of, you know, everyone, of course, this is to their advantage because then people would use Bruker as well. To answer your first question, I think that what we need to do is to work at the strain level. Of course, we work with spectra, and the spectra need to be perfectly good and so on. But if the strain is changing its name, then it is the strain that will change the name of the spectra, if you want. So there should be a kind of a chain reaction. And the basic unit is always the strain. And so this is what we are doing with other things like sequences as well. You know, we link the strains to, of course, we link sequences to strains and strains to species. And if the strain is changing for whatever reason, the taxonomic change or whatever, then, of course, all the things are changing automatically as a chain reaction.
yes.
Maybe there are some more questions, Marta?
Yes, first there are some people who are motivated to collaborate, so I will just read their comments. There is Cledir Santos. In any case, we are available to participate in this. Regards from Chile. And also Perrine Portier, who is saying we're developing a MALDI database for plant pathogenic bacteria, and we'll be happy to share our data in public initiatives. And then there is a question from Fortunato Palma Esposito, who is saying, are there true standardized protocols available for the analysis of all kinds of microorganisms, such as gram-negative, gram-positive, for example, streptomyces, yeast, fungi, in order to reduce the variability of the spectra?
Just for
There are standardized procedures, of course, but we have the knowledge already that there's not one single procedure that can work for all. We know for fungi, it's very difficult. Also for streptomyces, for instance, the ones that are performing that or producing the mycelium, it's difficult. But for gram-negative and gram-positive, which we use quite often, the basic ones in food, environmental, and in the marine-related study, the standard procedure works. But for instance, for rhizobia, which are known to produce those exopolysaccharides, we have an additional encounter to remove those first before we can start with the extraction protocol. It has an implication. For instance, you can choose to grow on agar plates or on broth medium, and for some bacteria, we see differences in mass spectra generated. So the same medium, but in broth or agar. So there is already a difference sometimes. It depends on what you are working with. And sadly, my colleagues at MUCL at IHGM, my apologies, will counter me with this one, Pierre. There are differences in their procedure compared to ours. Try to standardize as much as possible.
Okay. So that means actually that, but I wanted it would turn it the other way around. Actually, if you do have a media in for which the strain produce like mucilages or thing like that, then the question is about that you will not have the proper a proper spectra to be used, or you will have a wrong identification.
Well, not necessarily a wrong identification. Most often you don't obtain an identification.
Okay.
Because the spectra are so poor of quality that you get a no match. And Bruker has encountered this in their algorithm for sure, that if there are no matching peaks, then you just get a very low log score, the red dots that you get in Bruker. I'm not sure what color they get in MIRRI. You don't have an identification. But normally we've seen in the past that media has an influence on the mass spectra generated, and not necessarily on the identification, the final identification. So you can build your database using a certain medium that you use, for instance, often in isolation studies. But be aware that if your bacterium does not grow well on a certain medium, then you get the poor quality profiles, or if you don't get a good extraction from it, and that is hampering the identification. Differences in media, and they grow well, most often you get an identification score, maybe a little bit lower. It depends. But yeah.
I have another question, because I'm a naive user of MALDI-TOF actually. Is that and because of the presentation of Sylvain, actually Sylvain focused on some very key proteins actually, and so the question would be that could we use the data available from in the database from different spectra to try to go back and to match with the potential genomes and try to find out what are the best markers? Is it thinkable as just from the bioinformatic side? Is it to do? Am I just dreaming, or?
No, it's absolutely possible. Yeah, there are software to deduce the expected size of peaks. I don't remember right now the name of this software tool, but you can from a genome extract all the protein coding sequences and then deduce the mass and the size in certain conditions, and then you can predict some peaks. And so and reversely you can also identify the peaks you see based on genomic data. So that's not a dream. This is a reality. That's how we identified our peaks.
It's a way. What I wanted to say in a way to expand your system almost systematically with as many species as possible.
Yeah.
Okay.
I think what you're saying. Sorry.
No, please.
No, I think if I understand correctly, what you're saying is that once a few, you could run MALDI-TOF on a few exemplars of a given species or group, and then if there are many genomes in public databases, you can use the match. I mean the predicted peaks, and try to find them in the genomes to see if they are specific or not to more based on more isolates of each species. So that's a very interesting approach. Yeah, you don't need the strains then. You don't need the sequences to deduce the profile.
Okay,
but
so
you can do the same kind of thing, using machine learning, for example, and then you could try to correlate, different types of peaks with different types of properties. So you could use different types of machine learning runs if you want to learn from what's in there. The thing is that, and this is always the same problem, we need to store all the information at the strain level. Any kind of information, not only, of course, MALDI profiles, but also physiological, morphological, ecological profiles, and things like that. And when we have all those data together, then we are strong.
Okay, so I actually had planned a very, very short technical break for those who need it. And so, but I think, but it was good. It was a good transition to go for what's going to be the next, the last part of today's webinar with the genomic session. And then actually, because we had the chance to get more easily genomic data today. So I don't know if we can stay online, but actually we had planned to start at 5:30. I know that some speakers are all there. It's more about people have been hanging around for just 2 hours. Let's say 1.5 to have the possibility to have just a small break, and then if you're okay with that, we start in 4 minutes. Okay.
Okay.
So yes, see you in a few minutes. Bye bye, Anlene, and see you soon.
Bye-bye.
Hello, Antonis.
Good
Nice to
morning.
see you.
Good
Good
afternoon.
morning. Good morning for you. Yes, all right.
Yeah, right.
Nice to see you again.
Great to see you. Thank you for inviting me.
Yeah, it's good to see. Yeah, thank you for coming.
So you can hear me
Early.
well, right?
That's perfect.
Okay, great.
I don't know if you're still online or not. I don't know actually.
If I am or if they are.
If we are actually, I don't know exactly.
Yeah, yes, you're still online.
Okay, so we have to be careful what we say.
Take care.
There's nothing bad I would say.
So that's
Anthony.
Hi, Vincent. Nice to see you.
Same for me.
to wait a little bit that other speakers arrive and connect, and then we'll start again.
Sure.
I thought, yeah, and we will go straight. There was a mistake in the agenda, and so we'll go straight after the free presentation to the question and answer session, and we'll finish the meeting. So there was a kind of 15 minutes gap. I don't know where it come from, and so we'll go through and directly. So that will also free you a little bit earlier for your full day. So. So I would just wait 30 seconds more, and we'll start again. Okay.
Should I try sharing my slides, or is it too early?
Maybe you should. Yes, at least the first slide should be ready. Will be ready this way.
Do I share my screen or a file?
So usually is it a PowerPoint or a PDF slide that you have?
It's a PowerPoint.
It's a PowerPoint. So you can open your PowerPoint on your computer, and then you do shares, and then you click on the button that says share screen, and then you select your presentation, and your presentation will be on.
It should work.
It should work.
It almost always worked, except once.
Yeah, I'm not seeing it.
It takes a little bit of time sometimes.
Perfect.
Great.
Good. Okay, perfect.
Thank you.
So we're going to start again with the last part of today's webinar, and with the genomics session. And I'm really pleased to have here Professor Antonis Rocas. And Dr. Antonis Rocas is Professor of Biological Science and Biomedical Informatics, and Director of the Vanderbilt Evolutionary Studies Initiative at Nashville in the USA. And he will speak today about evolutionary genomics as a tool for studying fungal diversity and as well for revising taxonomy. So please, Antonis, the floor is yours.
Thank you so much, Jean-Luc, and thank you everyone for attending and for the invitation. Good morning from the southeast of the United States. I know it's good afternoon for many of you, but it's a pleasure to be speaking today. And so I'm an evolutionary biologist, and so our approach and our utilization of genomics is sort of tainted by our interest in evolutionary projects. And so what I'm planning to do in the next 25 minutes or so is introduce our project, emphasize a little bit the computational evolutionary approaches that we've used for studying, what we think is a major branch of the tree of life, and some of the interesting challenges we've encountered. And then also I'll give you a vignette about how we can use evolutionary genomics as a way to sort of give us insights into revising the taxonomy of our group of interest. And so, as I said, right, I'm an evolutionary biologist, and I analyze genomic data, and I'm interested in connecting genomic variation to phenotypic variation. And so this is a wonderful quote that I think is sort of knowledge to you already, that, right, the genome has lots of types of information. It's, you know, in some ways you can view it as a fossil record, you can view it as an indicator of geography, you can, you know, use it as a indicator of diversity, and so on. And it's evolved over the last few billion years, and so you can, you know, you can mine it for interesting evolutionary stories. And so the project we're involved with, and the project that I want to tell you about, is a project focusing on budding yeast, and I'll explain in a minute what budding yeasts are. And so the idea was to sequence the genomes of one representative, typically a type strain, of all 1,200 known species of budding yeast, construct their definitive phylogeny and time tree, and use that knowledge of phylogeny to revise their taxonomy. And then, given that yeast, sort of their lifestyle is largely based on metabolism, to examine the impact of metabolic diversity on yeast diversification. So can we map sort of changes in metabolism, changes in pathways involved in metabolism, to lineages that are species rich or lineages that are species poor, and how that goes. Now, I should emphasize, this is not just my work. It takes a team, and the team that has led this project for the last 10 or so years now is Chris Ittinger's lab at the University of Wisconsin-Madison. We've benefited tremendously from Clete Kurtzman expertise. I think it's fair to say he was the father of yeast taxonomy. Clete unfortunately passed away in 2017, and so we were lucky that, you know, by that time he was collaborating with Marie-Zeth Kronwald at the Westerdijk Fungal Biodiversity Institute, and so Marie-Zeth has become a key integral part of our team nowadays. And then my lab at Vanderbilt University. And you can see sort of under the titles, sort of the ways we pitch ourselves to the funding agencies. So Chris sort of has the genetics, genomics component, the computational evolution, and then Clete and Marie-Zeth are the taxonomists, but in reality, we're much more integrated than that. Now, of course, none of these people does the work. The work is done by, you know, a tremendous team of graduate students and postdocs, and it's really a delight to be working with all of these people, and even greater delight to see that many of them are now running their labs. Of the 8 people that you see, 4 already have their own labs and faculty positions in Asia and the US, and, you know, hopefully some point soon in Europe as well. So at this point, you may be wondering, you know, why budding yeast? What's the interest in budding yeast? So I want to spend a few minutes sort of telling you a little bit about their diversity. First of all, right, yeasts are fungi that reproduce asexually by budding or fission, and that results in growth that's comprised mainly of single cells. And so the yeast lifestyle is the lifestyle of unicellularity. So these fungi preferentially live as single cells. Now, this lifestyle has evolved multiple times in the evolution of fungi. And so if we take this fungal phylogeny, and you may be familiar with Cryptococcus yeast. It's one of the common and major human pathogens, especially of immunocompromised individuals, and they belong to the Basidiomycetes. There is another group of yeast called Malassezia, and they're responsible for many of the skin infections and dandruff. And they are an independent evolution of the yeast lifestyle, also in the Basidiomycetes phylum. And then there are the fission yeast, most known from Schizosaccharomyces pombe, a model for cell biology and the study of cell cycle. And they are an independently evolved group in the phylum of Ascomycetes. But, you know, my main interests are the budding yeast. So these include 1,200 species, including Saccharomyces cerevisiae, the baker's yeast, and Candida albicans, the well-known human pathogen that's responsible for several types of yeast infections. One appeal, especially given that, you know, we're doing this project in the context of 2-3 different labs, for taking on genomics of an entire lineage, is that budding yeasts have very, very small genomes. And so if you map the genome size of several different groups of organisms, you see that budding yeast are sort of borderline, you know, on the lower end of the spectrum of genomic size diversity of eukaryotes. And so they typically have genomes of about 10 to 20 megabases. And so that means that with technologies like Illumina pair-dense sequencing, we can go and get pretty good draft quality assemblies. Now, they're sort of morphologically, they're not that different, but I want to emphasize that they have amazing genomic diversity. And so if we take Saccharomyces cerevisiae, the baker's yeast, and compare it pairwise, compare its protein sequences pairwise with those of its close relatives in the budding yeast lineage, and we do the same for human and their relatives, all the different animal phyla, and we do the same for Arabidopsis and other members of the green plant lineage, what we see is that yeast harbor genomic or evolutionary sequence diversity on par with that seen in animals and plants, even though, right, all yeast, you would say, maybe look very similar to each other, but underneath it, there is extensive genomic diversity. And we can see that also not just in comparisons of yeast versus organisms from other lineages, but even comparing budding yeast with filamentous fungi. So here is a phylogeny that we built from 1,100 species of fungi. The budding yeast are about 300 species shown here. There are another 700-800 species of filamentous fungi in the Ascomycota phylum, and you can see that the branch lengths, which correspond to numbers of substitutions per site in our amino acid sequence alignment, are much longer for the budding yeast than for filamentous fungi. So you can see some of these characteristics. The genome size is smaller in budding yeast relative to filamentous fungi. Our evolutionary rate in budding yeast is much faster and slower in filamentous fungi. And then there are also other genomic differences, like differences in the GC content. Together with that genomic diversity comes what I would call genomic flexibility, which is definitely in contrast. For those of you studying bacteria or viruses, this won't be a surprise, but for those of you studying animals and plants, perhaps it's worth emphasizing. So if we just focus on the genus Saccharomyces, there are a few different species, Saccharomyces cerevisiae and its relatives. And if we do sort of the same sequence comparison of Saccharomyces cerevisiae and Saccharomyces eubayanus, they show on average about 85% sequence similarity in their proteins. And that sequence similarity is on par with the sequence similarity that we see between human and chicken genomes when we compare human and chicken proteomes. But of course, Saccharomyces cerevisiae and Saccharomyces eubayanus can hybridize, and they have hybridized. We have a species known as Saccharomyces pastorianus. That's the cornerstone of the brewing industry for lager beers. And it's a really interesting hybridization because Saccharomyces cerevisiae prefers to grow on warm temperatures. Saccharomyces eubayanus was originally found at very high altitudes. And the innovation of this hybrid, if you like, is that it's able to ferment ethanol at the pace that cerevisiae can, but can also do it at cold temperatures. And so this hybrid sort of shows mixtures of the two parental species. And this is the type of flexibility that, of course, right, you don't see in the mammalian lineage, you don't see it in vertebrates, you don't see it in animals or plants as much. Now, together with the genomic diversity, budding yeasts also vary widely in their diets. And so we have different species occupying all kinds of different niches. And so we have species that are nectar loving, and they live on on feeding off the nectar of different flowers. We have yeast that live in the Sonoran Desert in Arizona and form really close associations with cacti in the desert, as well as with the Drosophila fruit flies that eat them. And so that's another sort of ecological niche. We have organisms such as Eremophysium, which is a known plant pathogen. I've already introduced Candida, and you're familiar with them as human pathogens. And then there are species that are oil producers like Lipomyces and Yaroya, and they're of great interest to the biotech and biofuels industry. And so quite a lot of diversity. And one advantage of the lineage is that, as I mentioned, Cleet was the father of yeast taxonomy, and together with a few other key collaborators, he literally wrote the book on yeast. And for every species of the 1,200 that were generating genomic data, we have metabolic growth data. And so there is a table in the yeast book that for each species indicates whether it grows on glucose, whether it grows on galactose, or whether it ferments glucose and galactose, whether it grows on several different media. And so we can sort of generate a database of qualitative growth data for all of these 1,200 species and have at the same time sort of the genomic information that we sequence as well as the phenotypic metabolic information. And Dana Opulente, postdoc in Chris's lab, has generated also quantitative growth data for about a third of these substrates. So we have for about 20 or so substrate quantitative growth curves on how fast or how slow and at what pace these different strains grow on these media. And so so far we've generated genomic data for about 250 species, and most of them are type strains. And so to sort of the earlier discussion about how you can connect sort of strains to species and so on, one approach we followed is to sort of make sure we have the type strains, so that for any additional strains that are being sequenced, we have sort of the comparison to the reference. And we've united them with we've united these 250 genomic, 250 genomes with another 100 or so publicly available genomes. And so we have sort of a data set that covers about 1/3 of yeast biodiversity. And we've made sure that we cover as much of the generic diversity, the diversity at the genus level as possible. And then we have the qualitative data on growth on the different substrates. And so with that data, we can build the genome scale phylogeny, and you'll see that I've removed the species name because you wouldn't be able to see them. But just to orient you, I talked about Saccharomyces, the baker's yeast, so they are there. There is a well-known whole genome duplication, and it's found it has affected this group of species. Candida albicans and their related pathogens are on the other side of the tree over here. I should emphasize the different colors correspond to the different major groups of budding yeast that we have. Brettanomyces is another group that's involved in the making of lambing beers. They're really tasty. Yaroya is one of these oil producers. And then I mentioned some of these nectar-loving yeast, and these some of these belong to this Wickerhamiella starmerella clade, or WS clade as we call it. And so we've been able to mine this data set. I don't want to focus too much on the biology of the system and the biological discoveries. So I'm just going to give you the punchlines and then talk about sort of the technology side of things. But we really believe that budding yeast are a superb system for understanding genome evolution, for studying genome evolution in eukaryotes. And some of the insights that we've made over the years is that we see a lot of losses, not just of metabolic pathways, but also of fundamental pathways like DNA repair pathways. So we have lineages that have lost quite a lot of components of DNA repair or cell cycle. And so we're beginning to think that this gene conservation that we think of for essential genes or pathways is almost never universal. We see a lot of loss of traits, metabolic traits, and of the associated metabolic pathways. And so we think that part of what generates diversity in budding yeast is the loss of different pathways rather than their gain. And then, of course, right, metabolic innovation. These yeasts have come up with new ways to eat new metabolites, and so this has occurred not only through gene duplication, but also through a few, relatively few, but highly impactful horizontal gene transfer events. And so we've been able to identify entire transfers of operants from bacteria into these yeasts. And what's nice is that by creating this data set, other folks have started taking advantage. So our collaborators in Portugal, including a very talented graduate student, Carla Gonçalves, I think now she just got her PhD. They have identified also, or have been able to independently verify horizontal gene transfer, including of additional bacteria and operants. Ken Wolf's group in Ireland has been able to study mating type switching in the different species across the subphylum and identify multiple reinventions of their reproductive system. And also, Ken has also been able to study the genetic code. Until very recently, yeast were the only known example of a different, using a different genetic code for their nuclear genome. There was a recent paper in eLife that identified a few candidates in the bacterial domain that may have also reassigned their codons and work with an altered genetic code. But suffice to say, this is a data set that has been utilized and is being utilized increasingly by community, which is great. We see this utilization of data also within our groups, and so different people in the group have been able to study all kinds of processes utilizing these data sets, such as Abby studying codon usage bias, other graduate students in my group being interested, and in Chris's group being interested in the evolution of the galactose metabolism pathway and identifying horizontal gene transfers and reconstructing their evolution. And then, other folks in the groups have been interested in more evolutionary or phylogenetic questions and have taken this approach and this data to reconstruct the phylogeny of a fungal phylum or the entire fungal kingdom. And so in the process of generating these data sets, we've had to come up with several different computational approaches for handling these genomic data and for conducting these evolutionary analysis. And we also encountered several different challenges. And I want to sort of briefly introduce them and spend a few minutes talking about them. The first, of course, is that once you start sequencing genomes en masse, and for a period of years we were doing that, we have to develop pipelines that make assembly and analysis of the genomic data quite easy. And so along these lines, we developed an in silico whole genome sequencer, an analyzer that basically you would give it the genome data from many different strains that we had sequenced, and it would do automatically quality control and error correction. It would assemble all these genomes with several different de novo genome assemblers. Some are in Illumina only, some are PacBio only, some are hybrid. It would do evaluation, either standalone for the genome at hand or compared to a reference genome. And then it would produce an evaluation report that would basically enable you to identify which is the best assembly for the data at hand. And what's nice about this pipeline is that you can also sort of come up with several different experimental designs. So do you want to do just Illumina paired N, or you want to do Illumina paired N and made pair sequencing, or you want to combine, nanopore and Illumina, and it would, it would make up some data and it would rank the experimental designs, and so that you would have a flavor of, what you're looking at. We also had to come, so once we generate the genomes, then we want to identify orthologs from these genomes and gene families and be able to infer the evolutionary histories of genes as well as of the strains at hand. And so we had to develop quite elaborate pipelines for identification of orthologous genes that try to identify, and there has been continuous tool development by our group in developing these methodologies. Out of this pipeline, we would get a data matrix where basically we would have all the genes that we put in our phylogenomic data matrix and the species. And so each column corresponds to a different gene. Each row corresponds to each one of the 350 or so species that we have. And whenever you see a particular cell black, or whenever you see sort of a darker shade, that indicates that these genes in our data matrix are really well sampled and found in all species. And these genes here on the other end are found in fewer species, but they're still quite prevalent. And we had a filter of having, for every gene, we needed to have it in at least 50% of the genomes in our data set in order to retain it. And then out of this, we would do sort of quite a lot of phylogenetic analysis to try to get insights into the phylogeny of the lineage. Now, one problem you run into quickly is that as soon as you have sort of data matrices that this reach, and so this particular data matrix is 2,400 genes and 332 species, and so the number of possible trees that you could organize the set of 332 species is larger than the number of known particles in the universe, and very high. So simply put, there is no way we can exhaustively examine every tree, every possible tree for this combination of species. We need to come up with heuristic strategies. Now, a lot of software development in phylogenetics in the last 30 years has focused on this problem. and they've come up with very nice but heuristic strategies for identifying optimal trees. And so as the sizes of the data sets are increasing, so does the pressure to develop software that can infer very, phylogenetic trees from very large data matrices quicker and quicker. And so one thing that we wanted to evaluate is speed and accuracy of phylogenomic software. And so there are several different tools like RaxML and PhyML and IQ-Tree. And so we launched a project to evaluate, if we had all these single gene alignments and we run them through all these different softwares, which one found the best tree and how often. And so when we do that, we find that, no method is perfect. And so if you focus on just this graph here, the y-axis indicates the frequency of reporting the highest likelihood score. So how many times a particular method found the highest likelihood for one particular tree. And then the different color codings indicate RaxML in the shades of red with 10 starting trees. So they're slightly different versions. Whenever you see the 10, it's a little bit more of an exhaustive method, and it seems to be performing better. And PhyML, IQ-Tree, and then FastTree. And so you can see that FastTree is very fast. Computationally, it's the fastest of all of them, but it's the least accurate. So almost never you found the best tree for a particular gene. In contrast, RaxML and IQ-Tree seem to perform the best, and they found approximately 3/4 of the time the best tree. But you notice that 25% of the time they didn't. And furthermore, this is sort of the overall summary. These are individual data sets that are published, phylogenomic data sets that we analyzed. And so you can see a lot of variation in terms of the performance of these algorithms. And so for some data sets, the algorithms or some of the algorithms are really good, and they almost always find the maximum likelihood tree. And then for others, the algorithms are really horrible, and they almost never find the right tree. And so accuracy is something that we have to really begin worrying about. Even more worryingly, we started noticing something really strange with our data matrices, and that is we could have a particular sequence alignment for a particular gene, and we couldn't replicate the results. So if we run the phylogenetic analysis 1 year ago, we would get one result, and we would run the same phylogenetic analysis a year later in the same cluster and everything. The same person would run the analysis, and he would get a different result. And so Xingjing Shen, the former postdoc of my group, started wondering what's going on here, and he developed an experimental pipeline to test this. And so we take an alignment, and we subjected to 2 different runs with exactly the same settings, right? It's exactly the same alignment, exactly the same software, whether it's RaxML or IQ-Tree, exactly the same model, exactly the same seed number of threads on the compute cluster, number of searches, other parameters, exactly the same. The only thing that differs is that we run the search 2 different times, and potentially different machines are involved. And then we would evaluate whether we find, the same result in terms of topology of the tree each time. And, what we found is that sometimes the results are reproducible. They cannot be reproduced. And so, again, if you focus on this top panel here, we have the results for IQ-Tree with the light color, for RaxML with a darker color, and you can see that 18% of gene trees cannot be reproduced when we run the analysis the same time. In RaxML, that percentage goes down to 9% for the summary of all the different data sets that we run. But again, if you focus on the individual squares, you'll see that in some data sets, reproducibility is not a problem at all, like in Aspergillacea or Caryophyllales. But in other data sets, it's a huge problem. The only solution to this problem is to run the analysis with exact same specifications in the exact same compute node with the exact same architecture. The minute you change the architecture of the node being used, you start changing the results. Now, luckily, this reproducibility seems to be affecting individual gene trees and doesn't seem to be affecting sort of analysis of concatenated data sets or analysis using the coalescent. And so we can still be fairly confident of our species phylogenies, at least at this level of data size that we have. But it really is a question for the future. As our data sets are getting better, by necessity, our methods are going to become faster and faster. And so by necessity, we'll have bigger issues of accuracy. And it'll be interesting to watch the space and see how it develops, but it's not going to go away. All right, and so in the last minute or so, I want to discuss how we use phylogenomics to revise taxonomy. This sort of is an approach that we borrowed from the bacterial literature. Parks and their colleagues in Phil Hugenholtz's group develop an approach that measures basically the relative evolutionary divergence. And so if at the root of your tree there is no divergence and at the tip there is a divergence of 1, we can go and for each individual node measure or calculate the evolutionary divergence and then compare how this evolutionary divergence at different nodes scales with taxonomic rank. And so when they do that in bacteria, basically they get nice distributions like these that say that, right, the relative evolutionary divergence for the species in the data set is at 1, right? So there is very little evolutionary divergence, whereas at the level of phylum, the relative evolutionary divergence is really huge. And ideally, if your taxonomy reflects evolutionary divergence, you should see this sort of nice overlapping set of distributions, right? So the distribution of species is more to the right as you look at it than the distribution of genera, than the distribution of families, and so on and so forth. So we wanted to apply this across the fungal kingdom, and in particular, we were interested in budding yeast because we know they have very high evolutionary rates, and we know they have been historically under-described. And so we did this analysis, and we generated these distributions at the genus, family, order level. And what we see is that, the entire group of budding yeast sort of belong to a single order, Saccharomycetales, but in terms of the relative evolutionary divergence, they are far more divergent than the typical order that you see in the rest of the fungi. In fact, Saccharomycetales have more evolutionary divergence, higher evolutionary divergence than most classes that we see in the fungi. And so we can use this as an approach, and we can play with different classification schemes and identify, what's the appropriate sort of level of genomic divergence for reclassifying this subphylum. And so where we're going is now we have a data set of all 1,200 or so species, and we're in the process of analyzing it. And I hope I've convinced you that, budding yeast are a really cool system for studying the evolution of eukaryotic genomes. And I'll stop there. I know I'm almost on time. And so I'll stop there and again acknowledge the teams that did the work, and in particular the people that did the work. So I'll stop there and thank you and take any questions you may have.
Thank you very much, Antonis, for this really impressive. This is the impressive work of the last, let's say, 10 years. But yes, it was really impressive work. So, Marta, is there any question for Antonis?
Thank you. Yes, there is one practical question from Prasun Kumar. Prasun is asking, I have a fungal mutant, a product of random mutagenesis DS plus UV. It grows very slowly, though its enzymatic activity is better. Its slow growth is problematic for our process. It grows on potato dextrose agar in the regular media we use to grow its parent strain. What approach should I take to know the growth limiting factor?
Oh, that's a tough one. Yeah, I mean, this is really an experimental question. Probably, I don't know. Yeah, sequence it and then start seeing. But it's
Yes.
That's a tough one.
Okay.
Do we have some more time?
Yes, maybe a short question, and then we'll go back to the talk of Sarah.
Thank you.
And then
Another one from Vincent Robert. I suppose that these strange behaviors of the algorithms are related to the rounding problems, or that somewhere in their use randomization, right? Also, are you sure that large phylogenomic trees can be properly represented with classical 2D representations?
Yeah, I thought this would be a short question. Yeah, so Vincent, you're right. These are the rounding problems. So when we talk to the developers of the software, they say, yeah, we know, we've been telling people for 10 years. But experimentalists, so the people that built these phylogenies and use them don't know them. And so, right, to make the algorithms work quickly, you have to round, and right, you have to make some algorithmic decisions on how fast you calculate the likelihoods and the different scores of the trees. And so that's exactly what's going on. And you're right that, right, as we get larger and larger phylogenomic data sets, we really have to account for both sort of the variation that we see for each gene family, and then the variation that we see sort of at the level of species or lineage or strain. And I'm open to ideas on how we can represent them, but you're right that it's sort of becoming. It's also visualization is also a challenge that we'll have to solve.
So I think we could speak a long time for this about the questions, but even I restrain myself. So I will even so I think Sylvain won't mind if we wait a little bit for his question, and we keep that for the later, and then so that we could move to Sarah's talk, and I hope that we could have a final discussion together on all these projects at the end. So thank you again, Antonis, and now I would like to introduce you Dr. Sarah Leber, and she is a full professor at University of Antwerp. I'm sorry because I'm not familiar with the English way of saying it. And she heads the Laboratory of Applied Microbiology and Biotechnology. Today, she will present the implementation of genomics for the characterization of lactobacillus diversity and taxonomy. So please, Sarah, the floor is yours.
You're muted.
Your microphone doesn't work. Can you please turn on your microphone?
I think I was still on.
Oh, thanks. Are you sharing your presentation yourself, or do you need us to share your presentation?
Can you hear me now?
Yes.
Okay, then I will share the screen. Sorry. Can you see the screen now?
Oh, not right now.
I'll check again.
I can share it for you if you want. Oh, no, it's working.
I think it's, sorry for this short introduction. Can you see it now? Then I will start.
Yeah, it's working.
Okay, so good afternoon, everyone. Thank you very much for this kind invitation. Also, thank you very much to the previous speaker for the nice introduction, because I will talk today about how we implement genomics for the characterization of lactobacillus diversity and also the revision of its taxonomy. I think, as most of you know, lactobacilli are one of the best known beneficial bacteria. They're also often used as probiotics. But I believe that they kind of received a little bit wrong attention, or only a fragmented attention, which is mostly focused on potential in dairy applications and gut health, while their application potential is much wider. And that's actually also the idea behind my ERC project which is called Lactobia. It's on advancing lactobacillus beneficial potential, where we want to actually overcome the current knowledge gap in its biology, and really explore applications outside the gut for human health, also applications for animal health. We want to help the design of better or novel fermented foods, which are non-dairy. We also believe that some lactobacilli have potential for plant protection, even hygienic applications and forensic applications. And we also believe that genomics and also the natural biodiversity that is present is really an important tool to overcome this knowledge gap. And then yesterday, the World Antimicrobial Awareness Week started, and I think in this week it's actually also important to highlight that lactobacilli also have the potential for the development of alternatives for antibiotics. Actually, the field more than 100 years ago, and Mechnikov was a Nobel Prize winner. He already strongly believed in the potential of lactobacilli and lactic acid bacteria for certain health-related applications. But then with the discovery of antibiotics by Alexander Fleming and also more the direction towards the chemical industry, it was somehow a little bit forgotten. And now since the years of 2000, I think we have this renewed interest in bio-based and microbial-based solutions. And that's also a little bit the idea behind my research lab and our research focus. But we also believe that we really need a better framework and biological framework to design better applications. And so they should be knowledge-driven. And if we have this better knowledge, we can come to better human food and biocide applications, but hopefully also to lactobacillus-based drugs. These are also the kind of questions we ask in our research team, is how can we select the best strains for specific applications? How can we improve our biological knowledge on lactobacilli? And then for the topic of today, also quite key is how can we leverage all these major advances in all these fancy tools like genomics and DNA and RNA sequencing and so on, and to really advance our knowledge. So maybe in contrast to this previous speaker, we don't only use comparative genomics, or we don't only use this phylogenomics. It's one of the approaches we follow because we actually would like to combine them with different approaches. So in my life, we complement it with what I sometimes call a targeted microbiome approach. So we, for example, also study the prevalence of lactobacillus species in different habitats and environments by using all these microbiome sequencing tools, so more at the ecology level. We also we do real experiments to better understand the habitat adaptation of lactobacilli, and we try to do it as a high throughput screen. And we also use genomics and genetics and biochemistry related tools to really identify beneficial molecules. And for example, novel antibiotics are one of our key research areas for this part of the research. Maybe to quickly give an overview on the natural biodiversity of lactobacilli. I already mentioned before that people mostly associate them with gut health. They are present in the gastrointestinal tract in different numbers. For example, they're more prevalent in the small intestine than in the colon. But that's not their main habitat or niche in the human body. Actually, they're really dominant in the female reproductive tract. But you can also find them on the skin and in the nose. And this is also a little bit related to my own research history. I actually did my PhD on lactobacilli in the gut, and then I moved on to a postdoc on lactobacilli in the vagina. When I started my own professorship, we also explored lactobacilli and probiotics for the skin and the nose, and also started to work on lactobacilli in fermented vegetables, but also in food and even in honeybees and different insects. So then when I had the opportunity to apply for an ERC grant, I thought, we see lactobacilli in so many different environments and habitats. Then there are different species prevalent in these different habitats, but the picture is not so clear. So I really wanted to, in a way, take a step back from all these more applied research questions and have a more helicopter view on lactobacilli, which still focus on two main habitats to explore their biodiversity. And the first habitats that we really study in detail are fermented foods, which in a way are kind of strange environments because they're really man-made environments. You can really push the ecosystem towards this dominance of specific lactobacillus species or genera, which is also nice if you want to replicate certain ecological findings in the lab. And then we also focus on the female reproductive tract because that's the natural habitat of lactobacilli in humans. And it's also very intriguing that humans are the only animals where lactobacilli are dominant. It's not the case in other mammals or even related monkeys. So it's really an important. These two habitats are key for us to ask these key questions on where do lactobacilli originate from in our evolutionary history, but also in the life of an individual. And then when taking these steps back, one of the first steps we took back was looking at all the available information in the literature, mining the NCBI for genome sequences, but also from publicly available data. But then also realizing that our biased picture on lactobacillus biodiversity is also because we have a biased number of samples. It should not be a surprise that, for example, the gut or human fecal samples are heavily sampled, while, for example, vaginal samples of healthy individuals were actually almost not available when we started our project. And it was the same for dairy. The cheese industry and the dairy industry is really big in the Benelux area. But fermented vegetables and other habitats for fermented foods, they were much less explored. So these are two examples, the ISALA project and the Fermapecus project are two examples of sampling studies where we decided, okay, it's important for us to start our own sampling campaign and collect our own data. And I want to quickly show this sampling campaign we started for my year C project on lactobacilli in healthy women in their vagina. And when we started this project, we thought, okay, it would be really nice to have a high number of samples to explore the natural biodiversity of lactobacilli, but how to convince or motivate women to donate an intimate sample just in the privacy of their own bathroom or own house. And we decided that it would be best to follow a citizen science approach and also to actually design the project in a nice way. And we have named the project ISALA after Isala van Diest, who is the first female doctor in Belgium. And we decided to, in addition to my own scientific goals, which were really focusing on mapping the lactobacillus biodiversity, that it was also important to have attention to societal goals and to couple that really to the scientific goals and to kind of create a platform also for to open certain taboos on women's health and vaginal health, but also, for example, to put women in science in the picture, or also the bioinformatics in the picture, that you need to analyze the natural biodiversity of lactobacilli. So we launched a sampling campaign last year in the middle of the start of the corona pandemic. And in the beginning, I thought to be very conservative and only wanted to have 200 women, which I thought was already quite complicated. But within 1 week, we had more than 6,000 women who wanted to participate. So actually, we had to stop the recruitment phase. And we decided to invite all women to donate and swap if they wanted to, because we thought, yeah, that would be really nice to have a large sampling pool of lactobacillus natural biodiversity. All women, they received the sampling kits, and they sent us back 2 swabs, one for vaginal DNA analysis, so for the vaginal microbiome profiling. Most of the samples were done by 16S amplicon sequencing, but also part done by shotgun sequencing. But we also included a pink swab, which was really dedicated for the culture of lactobacillus isolates, so also to increase our culture collection of lactobacilli. So I don't have time to go into detail to the whole data that we have collected so far, but I'm happy that we managed, even during the corona pandemic, to within 1 year to analyze all the more than 3,300 samples we received from the blue swabs for the vaginal microbiome profiles. And we, for my talk of today, it's crucial to say, or key to say, that we indeed most of the samples were dominated by lactobacilli. And most of these samples were actually dominated by Lactobacillus crispatus and Lactobacillus iners, with also Lactobacillus jensenii and gasseri quite prevalent. So just a side note, it was a citizen science project, so then it's also important that you involve your participants well and that you also communicate well to your participants. So we also promised them all that they would receive their own vaginal profile and with also some information for lay people on the dominant bacteria. So here's an example of the pictures we gave to the participants with some information on background microbial information on how this bacterium look like and what science already knows about this. But for the talk of today, I think on the natural biodiversity of Lactobacilli, it's important to say that we have this sequence database, but also this culture collection and this novel biobank of Lactobacilli. And we add that to all the other Lactobacilli we have already in my lab, isolated from different projects and also by different PhD students and technicians. And I must say that, of course, the culture collection, it goes much slower than the DNA-based analysis. I think we are now at around 500 isolates that have already been properly identified, and this number is continuously increasing. And we are also planning, or we would love to genome sequence them all, because for us this genomic approach is quite crucial in our analysis. I hope the prices will still decrease a bit so that we will be able to sequence all these thousands of isolates. This is just a quick overview of the isolates we have so far from this ISALA project, and we're continuously increasing that. And we really would like to use this genomic information to better understand how Lactobacilli adapt to the vagina, but also to other environments, and also then to really identify the key beneficial molecules. But then I was talking quite a long time on all the efforts we did to collect our own samples, while it's important that in the database there is already so much information available on Lactobacilli. And we're actually also quite lucky that it's the most popular family from non-pathogenic bacteria, and there's really a lot of publicly available information available. And when you do this analysis, it's also important to include the publicly available information in your analysis. And here I want to quickly mention the work done by my bioinformatician Stan Witzuk in the lab. He is really doing all this more phylogenomics work. He did his PhD this year on the evolutionary genomics of Lactobacilli. And for example, one thing he did for his PhD was downloading all the publicly available genomes of the order of the Lactobacilli, so including both the family of the Lactobacilli, but also some more pathogenic families of the Streptococci, for example. And he also decided to do proper quality control filtering, and then also to make dereplicated data sets of maximum 100 genomes per species. And this pan-genome is really now at the core of our phylogenomic analysis. And he also made it scalable so that not everybody that wants to use it needs a supercomputer, but you're just able to run it on your own server. We're still doing the analysis, and I don't have time to go into detail of what comes out of this analysis. But for my talk of today, I want to quickly highlight as actually the side project we had to take to do this phylogenomic work, and it was the revision of the Lactobacillus taxonomy. Now, I'm not a taxonomist at all, but me and my team, we were stumbled upon the fact that the taxonomy of the old Lactobacillus genus complex was really a mess. And it was actually, at a certain moment, it was for us almost impossible to communicate well about the bacteria we were working with. And in fact, taxonomy should actually help you classifying and arranging the diversity of bacteria in groups with meaningful hypotheses and so on. And so they should also better reflect the evolutionary relatedness. So in a way, we were forced to go into the direction of taxonomy and to quickly show some of these problems. For example, in the tree of the Lactobacillus genus, there was actually also the other genus Pediococcus was present here in the red box, and also a whole family of the Leuconostaceae. And also we were a little bit similar as for the fungal diversity mentioned previously. We also stumbled with or had problems with this extreme diversity of the genus Lactobacillus in terms of sequence diversity, but also gene content and also in terms of ecological and metabolic diversity. And we were not the only ones. I think in the whole field, everybody knew that the taxonomy had to be revised, but no one was really willing to do so. Also because I think in a way also because of the industrial relevance of lactobacilli. And then so this international group of scientists, we came together and we decided, okay, it is a mess and it's a complicated task, but let's do it together. And also let's try to agree among the revised taxonomy so that at least people will, in a way, be willing to also use this revised taxonomy. And we also became involved because my team and with Stan as the main bioinformatician, we wrote this opinion in applied and environmental microbiology that we really wanted to have a genome-based reclassification of the genus. So this resulted in this taxonomy paper published last year, and on these taxonomic notes in an International Journal of Systematic and Evolutionary Microbiology. I also want to quickly show this infographic to show that the taxonomy revision was really done with only scientists and based on scientific criteria and based on a lot of phylogenomic work. But we also kept in mind that a lot of industry use lactobacilli in their products. And we also made an extra effort to communicate well to industry. And for example, I'm affiliated to the International Scientific Association of Pro and Prebiotics, and we also developed these infographics to also explain well how it is the revision of the taxonomy and why we had to do it. And so, how did we do that, and what were our criteria? So monophyly was important for us, that the new genera would be monophyletic groups, also that the diversity would be more acceptable, and that also the new genera would have defining characteristics. And then here, this tree is then important. This is a core genome phylogenetic tree of the Lactobacilliaceae. This tree was made by Michael Gensel and his team based on 114 core genes. While my team and Stan, we did the same, but we used almost 300 core genes, and we ended up with the same phylogenetic group, so a first indication of the reproducibility. So all the different colors are the proposals for the new genera. And at the outer layer, you can also see that we already double-checked that it made sense from a lifestyle perspective. We also checked that the new genera had a diversity that was more acceptable or more in line with related genera, and also that we also checked the criteria on exclusivity, so that the intragenous core amino acid identity was larger than intergenous. And then the third criterion was based on that defining characteristics. And I want to quickly mention one criterion, which was based on signature genes, which is a genome-based strategy, but independent of annotation. And we rationalize that signature genes are genes that are present in all genomes of a genus, but in none of the genomes outside of that genus. And we use these signature genes to really define, so at certain moments we were still doubting whether it was a novel genus or not. And then we use this signature gene criterion. And it's maybe better to show this on the figure. For example, all the dark blue columns are examples of signature genes, so they are only present in a certain genus, and we rationalize that they reflect a common evolutionary history or a common lifestyle, while the function of many of these signature genes still need to be identified. We also agree that some could result from horizontal gene transfer, but we could correct for that by sorting on the y-axis in the order of the tree. So as a result, we split the Lactobacillus genus in 25 genera, and 23 of them are novel genera. And I think for the talk of today, I think it's important to mention that the Lactobacillus genus, that state Lactobacillus, is Many of them are the vaginal lactobacilli, and then we also had to come up with novel names. So now, I said before, I'm not a taxonomist, so the novel names were designed by the taxonomists and Elisa Salvetti, Giovanna Felis, and Michael Gensel. They tried to propose names that started with an L and also that reflect more the ecological niche or habitat of the different proposed genera. But of course, that was not always possible, and sometimes we had to be quite creative to find novel names. And sometimes we also honored famous scientists in the field. So then to come to the end of my talk, and what can you now do with all this genomic information and the revised taxonomy? I want to end with some examples that it's really now more practical, although maybe the new names don't sound that practical. And thanks to this genomics and taxonomy, we can really now come to new insights in the evolutionary history and habitat adaptation of lactobacilli. And again, in my lab, we focus now on the vagina and fermented vegetable as main habitats. And this is, for example, an example of the microbiome studies we did in this ISALA project with more than 3,000 samples that have already been characterized and information sent to the participants. With the old taxonomy, we just say they are all dominated by lactobacillus species, while with the new taxonomy, for us, it was actually quite surprising to see that the second most dominant genus is the Limosi lactobacillus, and it's actually a new taxon we are really intrigued by to study further in the vaginal niche. And then on work on fermented vegetables is also being continued, and at the top you can see the old taxonomy that we were not really able to capture the diversity very well. While at the bottom, you can see that we are now better able to capture the diversity and the different taxa that can be present in fermented vegetables. So that leads me to my take-home message. I think thanks to the implementation of genomics for the characterization of the lactobacillus diversity and taxonomy, we can now really start our work and start asking questions we could never answer before or ask before. For example, one of the fundamental questions related to applications we ask ourselves is, does the interaction of certain nomadic species differ from interaction with more host-adapted lactobacillus species? And a second main question we have is that, for example, we find this lactobacillus taxa in the vagina, and they're also, in a way, rather evolutionarily closely related to the yogurt bacteria. And we wonder why that is the case, but a more maybe fundamental level, we then also ask ourselves questions: when did key events in evolution happen? Why are lactobacilli rod-shaped? Why have they lost their sporulation genes? Why are they why did they start to focus on lactic acid fermentation? Why are so many lactobacilli anti-inflammatory? How do they interact with the immune system? And we really use the genomic information for this. So with that, I would like to acknowledge all the people involved. I really would like to thank my taxonomy collaborators. It was really a big task to have to revise the taxonomy, to have this over international meetings already via Zoom, even before the pandemic. But I think we are all quite happy with the end result, and we are very happy that the results still hold true. But from more the practical point of view, I would also like to thank my ISALA team for the sampling campaign on the biodiversity of lactobacilli in the vagina. And the same holds true for the sampling team for the lactobacilli in fermented foods. I also would like to thank them. And I would like to thank the funding agency, ERC and FW for help with this.
Thank you, Sarah, for this very interesting and impressive work on the importance of lactobacilli, especially as well for vegetables and human health. And I hope I'm sure that Marta, your work has raised a question.
Well, there is no questions
Not
for
yet.
now, but yes,
Not yet.
but I invite the people to ask the questions, and then we can ask them during the Q&A session. But there is one very positive comment from Matteo Schwilk that I would like to join as well. He said, "An absolutely amazing, brave, and important project. Congratulations."
Thank you.
I was wondering, you mentioned that you are collecting a huge amount of genomes from the databases. About, you, I don't remember, it's about 20 or 20,000 genomes.
24, yeah, we.
And how much did you deduplicated the genomes? I mean, on which criteria did you duplicate? I mean, you wanted to have less than, let's say, 0.1 thousand diversity maximum or something like that for the whole genome. 0.1% not 0.01%, something like that. I mean, what criteria did you use to simplify? And the second question is, what type of equipment did you use? You mentioned that you're able to use it on simple computer.
Yeah. So the exact details, I invite you to contact Stan Wittek, who is my bioinformatician. But we wanted to dereplicate at the end to have 100 genomes per species and then per genus.
Yes.
To have a little bit, to not be biased by this. And he also really wanted to have a scalable tool, so that it would not only be usable by people be able to run it on the supercomputer, but also on our server. And he really managed to have this scripts written, and it's really scalable and usable in our group. And then you can really choose the rank of your taxonomic rank of your interest to study certain evolutionary questions.
And you mentioned, I'm sorry, you mentioned 100 strains per species or genus, which means what is the size of the overall set?
The idea was to start with 100 strains per genus, but then of course for many genera there were not enough available. So then he decided, I think the most of the work was done with 100 species per genus, and then in this way, and then it was available for most of them. And then of course he always started with the type strains and double check those, but then for many, and in that way he built up the tree. But I have to, the work is still, it's published in his PhD thesis, and we still have to finish up the work and publish it. And so indeed, but it's our good, good
just
points
to know, just to know the size of the sampling that you have to handle. Actually,
for that we didn't sample. We really downloaded all the 24,000 available.
The genomics sample of data.
Sampling from the database, yeah.
Okay. So, do we have other question? No, nothing else. So, okay, let's wait for the final question and answers part after the talk of Federica. And so, thank you again, Sarah. And so now we have our last talk of the day, and from Federica Palma. She's a project manager at the Biological Resource Center of Institut Pasteur in Paris too, and she's coordinating the development of a system for genomic taxonomy. And she will speak us about the use of genomic data to catalog and analyze strains biodiversity within bacterial species. So now, Federica, it's your turn.
Thank you very much, Luke, for your introduction and presentation and invitation also. And also thank you to the previous speakers that greatly introduced the importance of this talk, as we saw that the many data and information that are generated for strain as metadata, phenotypic data, and genome data need to be integrated, stored, and organized in cataloged in usable way, and also through maybe practical interface. So I will give you a talk. My talk, I will give you an introduction about what genomic libraries are, how they are built, and why we need to build them. And also will give you some information about our genomic taxonomy platform, And an example of one of the database that we host in our platform to finish with some conclusion and perspective for the microbial biological resource centers. So what is a genomic library? It's a collection of strain variants which can be described for a specific bacterial population, for a bacterial species. And genomic libraries includes data from the isolates, such as the metadata, provenance data, time of isolation, country of isolation, and it can also include relevant phenotypic information and, of course, genomic sequence data. And why do we build this genomic library? Is to catalog and analyze the huge biodiversity that we have of strains that we have within a bacterial species. And as you know, and as we saw during this webinar, bacterial species are composed by very diverse strain, by a galaxy of strain that can present very different phenotypes, and these phenotypes can both have interest in terms of clinical or in the food industry. As an example here, we can see, let's say, for the species, the Salmonella enterica, that we have hundreds of sublineages within the species and thousands of genotypes, which are the sequence types, that can be associated to particular phenotypes, such as the clinical outcomes. And during time, these strains from these species have given names to differentiate them for the other strains of the same species. And often these names has been based on the Latin binomial nomenclature system, that is the linear nomenclature, that anyway not always capture the diversity of the species, such as represented in phylogenomic reconstruction. So this brings us to a big challenge, which is of the strain taxonomy. As the linear taxonomy may well answer to specific questions about phylum and classification of phylum and species, but we enter in what Sylvain likes call the Babelian zone, when we need higher discrimination for the epidemiological studies, for example, to discriminate clone of a species or also individual isolate. And how to tackle this challenge? Thanks to the next generation sequencing, we can improve and increase the discriminatory power of identification and molecular typing, improving also the characterization of isolates through the application of different approaches, such as the multi-locum sequence typing approach that can be also, thanks to the next generation sequencing, extended to the whole genome, so to the core genome or whole genome multi-locus sequence typing, and also to another approach would be also the whole genome single nucleotide polymorphism analysis. But what multi-locus sequence typing that was developed in 1998, also if it provides high reproducibility and characterization of isolate, anyway, it lacked of discrimination. So the advanced in the whole genome sequencing and the development of tools for genotyping have in fact provide us a very high level of discrimination. And how does this tools works for who is less familiar? This approach, which is also called gene by gene approach, relies in fact on the analysis of the loci which are distributed around the genomes, 7 usually for the case of multi-locus sequence typing, but can reach 100 or 1,000 for core genome MLST. And how does it work? The approach, in fact, once the sequence is detected, it can be translated in a numeric identifier, what we can see here, and all the variants of allele for this locus can be assigned with a specific identifier, and the combination of such identifier, which are inside the profile, can be translated in a sequence type that that it's really more easy to compute and is more scalable compared to other approach. And what is also the advantage of this approach is that novel allele profile, when appear, can be defined over time, and that this profile, in fact, can be quickly handled and analyzed from a computational point of view. So what is the potential of the genomic libraries? So we said before, it can be really essential for taxonomic, phylogenetic, but also for evolutionary research, genetic diversity, and the surveillance of global spread of circulating strain of interest, but also to comprehensively decipher the characteristic of concern, of interest of bacterial strains, such as can be antimicrobial resistance, virulence, or maybe some technologically interesting future. And also all this information is needed to explore the possible association between genotype and phenotype, and also to answer to a variety of questions. But how do we build this genomic library? So we need to collect genomic resources that, as we saw, are largely available today, or we may need to generate new one for ideally capturing all the natural diversity that can be present in bacterial species in a population. And when pair hand read sequencing strategies are employed, this can the high quality bacterial genomes can be assembled. And once they've been assembled, these sequences can also be annotated and curated, comparing with database of genes. And the genomic data, the provenance data, phenotypic data, must be integrated and also stored and organized, but interpreted and why not disseminated in a usable way. So all of these steps required a sustainable infrastructure and need to be achieved with in line with appropriate standards of accuracy and openness, while meeting the ethical and legal requirements for confidentiality and also data ownership. So all of these can be a successful example of this kind of infrastructure. It's represented by BixDB Pasteur, which is an infrastructure coordinated at the Institut Pasteur Microbial Biological Resource Center. This is a platform which is powered by the bacterial isolate genome sequence database software, which was developed at the University of Oxford. This platform is running since 2017 in Pasteur under the bixdb.pasteur.fr domain. And it's really a successful example of integrated resource of genomic libraries and bioinformatics tools that enable the storing, sharing, and comparison of data. This platform serves, in fact, as a one-stop platform for bacteria strain classification and taxonomy, and that allow global laboratories and public health institutions to communicate on strain subtypes with a common language, but also to better track the emerging bacterial strains in across domains, so in a One Health or Global Health contest. In this schema, I reported a bit the design of the database, where in one side we can see the isolate genomes metadata and the result integrated from the genomic analysis. And on the other side, in the center, here we can see the allele and profiles, which are cataloged to allow us to provide a nomenclature service that basically consists in the translation of the allele sequences in this ID, as explained you before. And it's fostered by the development of schema from our research group, and also it is maintained and curated by expert team of curators. And for sure, which can be specific for some bacterial species of clinical interest, such as Klebsiella and Listeria. And also these databases are contributed by thousands of users. And the core function of this infrastructure are so the collection of data, which include the quick and easy upload of public or private data through an automatic system, and then the validation of this data and the feedback provided by the curators, and the nomenclature service when new variants need to be designed, and also the communication, standardized communication between users and submitters and curators. And last but not least, it also provides many tools and plugins for visualization, export of data in standard format, and also sharing of data of projects inside the platform. So genomic libraries in BigZB Pasteur are dynamic resources that constantly expand as these actors and members of the platform interact between themselves through submission of genomes, or through of public access, or also through submission of private data, while the team of curators basically integrate, curate, and maintain updated and of high quality database. So to give you an overview of the platform, it hosts several species databases with more than 20,000 isolate for a really a number of countries, like more than 150 countries, and it includes an incredible number of alleles that have been defined, stored in the database, and organized in the several 40, I would say, but more than 40 typing schemes. Also, it includes a very high number of genomes of high quality, and as I told you, in private spaces that mainly include a really high number of genomes. All this data, as I told you, can be analyzed thanks to the services and the web tools that are available from the database, such as the sequence typing tool, the quality check, phylogeny, and also can be easily queried through our application programming interface functionalities. So the database is largely used. We can see we have plenty of visitors each day, more than 1,000 registered used, and we receive more than 100 curation for months. Here I want to just give you an example of the Klebsiella sequence typing database, where it looks like this. It can be accessible at this link. And you will see here you can find the sequence typing databases, and also the information for the data access and upload, and all the information about the software. This part we can find the curator interface databases. And here I wanted to just remind that in this database we analyze the Klebsiella pneumonia species complex in particular. And about this species complex, we collected more than 10,000 strains, most of them with a genome associated and minimal requirement for provenance and phenotypic data, typing data. These data are from around 120 countries and allow us to define around 4,000 sequence types. And the contribution of this data has been given by hundreds of data providers and allow us to develop around 20 typing schemes for this genus, for this species, sorry. So the implementation strategy, again, to remind you that the platform include user-centered analytical workflow, which integrated the genomic taxonomy for surveillance and detection across sector and cross-national borders. The platform is fully open source and open access, and allow programmatic submission of data, and it's also interoperable with existing systems to enable the adoption of an harmonized nomenclature, and also for automatic exchange of data. In addition, it also ensures a secure network, confidentiality of data, and ownership of data, and allow for the development of standardized network frameworks. So all of these provide the platform publicly accessible population-wide information, which are described with a uniform nomenclature and linked to provenance and phenotype information that allow for several outcomes, as we can see here, such as typing and phylogeny, and also for monitoring, for example, of vaccine implementation or antibiotic resistance, and much more. So here I stole this illustration from this important paper from our developer at the University of Oxford, that I think it really captured the relationships which the variety of relationships which are supported by such kind of genomic library platforms. In fact, where we can see that there is a large community of and diversified community, which may need different methods to answer a specific task, and also a variety of tools that need to answer the needs. And what is most important is that these tools are available through a graphical interface. So even for lab where the bioinformatics skills are limited, there is the possibility to still analyze their data and find meaningful information. So to conclude, it is clear that WGS approaches provide a number of practical advantages in addition to its inherent high resolution and comparability of nomenclature and with the indexing of bacterial diversity. And with a single test here, it is possible to eliminate maybe different phenotypic tests that would be more time consuming, and also with a single test we would enable the characterization of multiple future in isolate that can have a clinical or technological biotechnological interest. So the genomic libraries in BigDB Pasteur are dynamic resources that can be constantly expanded thanks to the contribution of our old user, of the new user, or thanks to new collaborations, all collaborations, and which ensure a uniform nomenclature of sequence data and the link of this data to relevant phenotype and provenance information through the user-friendly interface, contributed to the plenty of studies that we saw before. So one important point for us is that to meet the specific need of each user, we also made available these private spaces and to allow to support more confidentiality and pre-publications and also the development of patents. Last but not least, the BigDB Pasteur infrastructure quality and longevity is ensured by the several group of international experts, including microbiologists, bioinformaticians, which are dedicated to the platform, and that constantly communicate between them and ensure communication with end users. That can be from very different institution background and so on. So what are finally the perspectives for the Microbial Biological Resource Center? The BigDB Pasteur infrastructure, it's open to mount new genomic libraries, establishing collaborative projects with the Microbial Resource Centers, which are interested in analyzing bacterial isolate from different sources. It can be environmental, industrial, food, or clinical, to aiming to improve the identification or also the molecular typing, the taxonomy, the study of biodiversity. In addition to the valorization of bacterial strain collections, such as with the characterization of future of interest that can be antimicrobials or bioactive compounds, ensuring the interoperability with already available system, these infrastructure, these genomic libraries can facilitate the detection of resource of interest to define some, for example, phenotypic properties of clinical or industrial purpose, thanks to the genomic data and the strain identifiers that were deposited in the genomic libraries from the Microbial Biological Resource Centers. So as take-home message, where libraries can help you, you and the Microbial Resource Center, the genomic libraries can organize and characterize the large amount of data where large collections are available, and also it can facilitate the location of relevant information and to spot out representative strains useful for preservation and distribution.
Federica?
See?
Sorry, I don't see your slides anymore, and other people also.
Really?
Yeah, since 1 or 2 minutes.
Oops.
And basically, we are thinking that you are reaching the end, so this is why I didn't want to stop you, but I would have done it.
Sorry, but you see me.
No, yes, but just see you, not the slide anymore. But it would be nice to conclude, basically. So if you don't,
I was really close.
It was a technical message, and then.
I don't know what's happened. Maybe because it passed 17. So here we are again. You can see now.
Yeah.
Okay. So this was the take-home message, and where in fact the most important is that this genomic library will allow you to enhance the accessibility and link to all the information from metadata, sequence, and phenotype for your favorite bug. So really to conclude, you will understand that we have several open access database, and if your favorite bugs is not featured, you can contact us and we can discuss about setting up one dedicated database for you. Just to conclude, I want to acknowledge all the person and the members that participated in these along several years on this project, especially the taxonomy group of the Bristol Lab, and also in Institut Pasteur, our colleagues from the bioinformatics department and from the high performance computing department, as well as I would like to thank our partner developer, the University of Oxford, and the World BixDB Pasteur team, CRBP team, and all the collaborators from now and from the past that contributed to that. And also I want to thank you for the attention. So I guess we can pass to the
Thank you,
question
Federica.
and be happy to answer.
Thank you, Federica. So, Marta, is there any question?
Hi, Jean-Luc. Well, there is a question that is hanging still the presentation of Antonis Rocas, so probably I could...
Yes, but maybe we just because it's a fungus-related question actually from a bacterial specialist, and so that it implies that Antonis answers. So before we should have any question to Federica, if any.
No, there is no other questions.
I have just one smaller question from my side, because in her slide she mentioned, and when she encodes the different genotypes that you mentioned, they could do that for bacteria and also fungi, as I remember at the very beginning, actually the way. And actually, I didn't see how you handle with heterozygosity, because technically you have at the same loci you may have the two possibilities actually. So I don't know.
Yeah, this is one of the challenges of our technique. We can have multiple allele for a single size. So usually we keep the consensus. But so for bacterial, it's not so. For the bacterial species that we analyze, usually it's not so often.
Yeah. No, but yes, it's because you mentioned fungi, which are eukaryotes and not always can be diploids. Then the question, and I'm working also with diploid organisms, basically.
Yeah, unfortunately, I have not.
This is obviously the same tricky question. Okay.
Yeah, I have not so big experience on that. What I could say for bacterial, it's that we may have this kind of situation where multiple allele are assigned to a single locus. So there are different ways to tackle this problem, and sometimes using the consensus between multiple within a large data set, or also keeping the lowest assigned identifier, for example. I don't know if Sylvain wants to add something about that.
Well, very briefly to answer the question of Jean-Luc, yes, we do have databases with diploids, and we use the degenerated base codes from UPAC to create alleles in these cases.
Yeah,
for Leishmania, for example.
Yeah. But if you do phylogenies, I'm not so sure it gives the same answer, actually, because this is what we have for yeast. And actually, the more it goes, and it gives... but then I would leave this opinion to Antonis, that who is much more... a much better specialist on these issues about phylogeny for eukaryotes, actually. And it would be just, I think, now the best time to go back to the forum and ask for about to discuss about all these issues. And one thing is connected to the discussion of Sylvain to Anthony that we have left, and then it's about the question of but please, Sylvain, you remember your question actually about ANI. So maybe it's better to ask you directly.
Trivial question about, we in bacterial species taxonomy, we define species using the average nucleotide identity threshold. And I was wondering how this goes with fungal species.
Yeah, that's a great question. So typically in fungi, if I was to say what is the most popular species concept or approach, we use MLST typing. So 3-4 loci, and we build phylogenies, and if they're congruent, we call that a member of a species. Some folks have started arguing for a taxogenomics approach. So basically, go ahead and sequence the genome of the type strain, and then we can have sort of genome scale phylogenies or genome scale analysis. We've begun, or we've for 30 different fungal species, we've collected data from GenBank, or all the available genomes from GenBank, and measured ANI. And typically, our approach was to make sure we remove misidentified isolates. For most species that we were confident these strains are part of species, they were sort of between 95 and 99% similar. And so I think it would translate pretty well. And definitely it's a great measure for detecting species misidentification or strain misidentification. I think previous speakers commented, right? GenBank has lots of misidentified genomes. And so that's sort of a good approach. And then your question about sort of the HGT part, luckily for fungal species, for most fungal species, the frequency of horizontal gene transfer is very small. And so, in a typical yeast genome, we can detect, say, 10 horizontal gene transfer genes, right? So if we examine all the genes for evidence of horizontal gene transfer, about 10, 15. It changes a little bit with industrial isolates, and it changes a little bit with, in certain cases, but it's not as big an issue as it is for bacteria, where, right, every gene could potentially be horizontally transferred. And so we have it quite a bit easier in that sense.
Thanks.
Thank you.
So we do not have any new question right now. I have Mike. I have a real. It's always it's clear for me that from all these talks today that we have, and because the cost of sequencing has dropped sharply, that it's getting more. We are getting more easily. We have more easily access to full genome sequencing for more and more strains, more and more data. But the question is that what I have one question for you is that what is the trend actually? Is it make it sense to sequence genomes as a fully genome, because that means a lot of information, and to stock to handle it, and or should we just stick well, which is already nice to type strain as a kind of tool for the exploration of diversity, and then use let's say more high throughput or tools like MALDI-TOF or for intra-population identification, characterization. Sorry. Basically, I have my own idea, but I want you to answer about that.
So who should answer?
All of you, please. Any of you.
Well, maybe I can start from the lactobacillus field.
Please,
I think it's really depends on your application, but I think for lactobacilli, when you want to apply a specific strain for a specific application, you really need a whole genome sequence, because one gene can really make a difference. And we have found in our own research that changing one gene can make lactobacillus a more pro-inflammatory or an anti-inflammatory strain. So that's always crucial. While from a more evolutionary point of view, the evolutionary genomics people in my team, they sometimes only need one genus per genome per genus, and then they have enough to recalculate more the evolutionary history. So it depends a little bit on the questions you ask.
So at Pasteur, in terms of quality control of strains, we tend to prefer genomic sequencing nowadays compared to MALDI-TOF, which has been demonstrated today also to be limited for within species diversity. I mean, most strains would have the same MALDI-TOF profile. So there is no, when you start manipulating many strains from the single species, there is no way that MALDI-TOF can help you make sure you are dealing with the right strain. And so genomic sequencing is obviously one of the approaches. And we tend to use what Federica just presented, core genome MLST, which is a highly discriminatory fingerprint for each strain, basically. And so that's our preferred approach when we can apply it.
Maybe the last, maybe Antonis will give his point of view about it.
Yeah, so I mean, it really depends. I agree with Sarah. It really depends on the question that you're asking. I mean, as an evolutionary biologist, I'll never say no to sequencing genomes, and I do think as you go to population level, the deeper the sequencing, as you know from your studies of the galactose pathway, the deeper you go to the sequencing, you start finding the rare variants and really interesting phenotypes that sequencing 10, 15 strains, you're not going to find. And so, adding, as we sequence more and more isolates or strains, adding the ecology bit is going to be where the really amazing insights are going to come from, in my opinion. And so, we've focused on building the framework at the species level, and then, it's clear the next question is going to be population genomics and connecting the ecology to the species. And, that's going to take, the next 10, 20, maybe more years, but it's going to be fascinating because, in some ways, the phylogenetic framework is step one, right? So you need a tree to sort of conceptualize the process. But then, then you start having the variation aspect of it and the diversity aspect of it and connecting genotype to phenotype. So my approach is, genomics. It's not worth doing any of the other techniques. But, if I was running a database or if I was running a culture collection, my approach would be different because cost is a real thing. Right?
There are if I may, there are two things: the cost of single strain sequencing, but also the amount of data that it generates and to handle. Actually, even for bacteria. Well, I do not say that bacteria have small genomes. Not what I wanted to say, but as soon as you go to eukaryotes, then the genome starts to be bigger, and that means a lot more information to stock, which means and to handle. I mean, for a small collection, it can be maybe handled more easily, but it grows very quickly, and it's really there's a time we have to adjust the the amount of information that we can handle. And so, but yes, basically, well, but Antonis knows that what I did. So you know that I'm keen on yeast variation. So I could not. I would also explore the full genome. So, but the handling is more about handling the data, having the tools to stock the data, and to analyze it, to visualize it, and to detect what we are looking for. And it's also depends on the question we have. I agree with you, all of you. Okay. So
if I add something, I totally agree with you that we lack the tools, but that's right. That's a funding priority issue, right? It's people don't want to pay for tools. They don't want to pay for databases, but they want them free. And so, right, as a community, we need to support these collections. We need to support the development of that infrastructure, because otherwise it'll be sort of, right, I make my pipeline and Sylvain makes his own pipeline and Federica makes her own pipeline, and it's the tragedy of pipelines.
And also sustainability long term.
Yes, and I was asking you, Federica, because you mentioned that you propose a space for users own analysis, but also it has a cost. I mean, if each one has its own space in your facility, that means you propose them space. I mean, if they are big users, they will use teras of data for their. You're maybe not exactly ready for accepting hundreds of genomes.
In fact, our basic philosophy is the open access and open data, but we know that many users maybe cannot, for reason of confidentiality, or maybe food industry cannot publish their data so easily. So so far we propose this space, and we are working to develop a business model to integrate to our platform for a user with really specific needs and big amount of data. As Vincent said before, if it's one, two, few isolate and few identification, few typing, it's okay. But when it starts to be hundreds of thousands of data, it starts to be more challenging and to require increasing computing resources and threads and core for analyzing data. So yeah, we are working on that.
Okay.
Yeah. But Jean-Luc, I would also, I would answer your remark by saying that storage is negligible in terms of costs. What costs is really the human power of analyzing and setting up things and curation, but really storing terabytes doesn't cost anything anymore. So it's not a big issue for us.
I agree with that. That a lot of work has to be done. How we have to pay for the. I have to build the structure, but also to create data. That's for sure. And same for the Vincent database for MALDI-TOF as well. But another point too is that if we put that in public database, then it's also and we download the data, make it available. Also, it's also it will, on a long-term basis, it also costs energy and so on. So it's not nothing. It's just all of this has a cost.
It's quite significant, when you look at the cost of hosting in data centers and so on. It's really expensive. And if you have a lot of calculations to do and so on, the CPU costs are really high. So you really need to think about that. That's really important for the future, I believe. And also, many of the software, if not most, if not almost all, are really limited in terms of scalability, and they can compare, thousands, maybe 50,000 or 100,000, but what will it be when we will have millions or tens of millions of people? And really, it's coming. It's not that it will be in 20 years. It will be there, and it's already there in some cases.
when you look to the genomics, in some years ago, people were sequencing 10 strands, then people moved to 100, and now we have 1,000 sets of strands from it's the 1,000 yeast genomes as an example, and it's almost systematic. I don't know how well. Sarah spoke about 24,000 strains, but she dereplicated, so she cheated a little bit.
But I would also say that, sorry, Silvan. The mutualization, I think that the way forward would be also mutualization of resources and interoperability between systems that would allow to avoid the duplication of data, and so the real-time usability of this data.
So, which means basically it's a kind of a link between MIRRI and Elixir, something like that, who may help to solve put together tools and develop together tools and maybe and mutualize efforts.
A few weeks ago, there was a symposium dedicated to that, to the handling of not thousands of data, of genomic data, but millions. And that was on the Royal Society website. You can look at the talks; they are still available. It was the symposium organized by Mark Hartman, Kat Holt, and David Ananson, and they discussed a lot of tools to do comparative genomics of millions of genomes. So it's quite interesting, and it's amazing to see how the field is evolving fast in terms of bioanalysis too.
If I may say something too, Jean-Luc, there is another point that we need to address is the reward of people working with databases, because many people that are entering data in databases are not rewarded at all for what they are doing. I see many curators that are entering data day in, day out, and very often they are not even mentioned in the publications or things like that, and they are still evaluated at some point by an index, an H-index or things like that, which is not capturing the information that they are entering in databases, which is very sad and unfair, I would say.
Yeah. Yes, and the quality of the information that is saved and curated is really essential for everything. So I have one in the question section. There was one comment from Matthias Wilk, which says it seems that there is an inflating multiverse of attractive database platforms which often contain redundant information and services. Can you see a future for a more united and coordinated approach? Although I now see that it's mostly a question of money. Well, it's not only a question of money; it's also a question of matter of federating the efforts. And so, does anyone want to comment that?
No.
It's very challenging, yeah?
Yes. Well,
it depends. I think the resources are typically, they need to be built to address a specific question. It's like the Unix philosophy. You have one common per question or per need. And for example, in population biology of bacterial pathogens, what we do is we tend to create genomic platforms for single species nowadays, or genera, because the questions and the way we can address them are kind of pathogen specific. The virulence factors are not the same, from a Klebsiella to a staph to a TB organism. And so it makes sense. And also the epidemiological questions are not the same. So it makes sense to focus these platforms so that they are really tailored to the needs of the communities. That's one of the aspects. And then, for example, if the question is how can we use genomes for reference identification, then you need a kind of universal database with, for example, ribosomal MLST or 16S, which really addresses this question. So there is no universal system, but you need always to match the answer with the question. I mean, the strategy with the question.
Strategy. Okay. So I think it's already 5:30, and I don't know. I think it's we are basically 5 minutes later than that we had planned. Even with without taking care of the break that was supposed to be taken. But anyway, it was a real pleasure to have you all and to discuss with you. And so I think we have to conclude for the day. I don't know if Luis is in here, but if he's not there, I will do something for him. So the first thing I have to say to thank you all for all speakers for today. You here for the genomic session and the MALDI-TOF session, but also all the speakers for this morning for the microbial section and the culturomics. And I have, in addition to all the speakers, and also we have to thank all the people who attend the meeting as well, and who have been the courage to wait until the end of the day. I have to thank all the person that helped me to help us to construct the program. I have a special thanks to Emmanuel Maguin for INRAE that helped me to contact people from the microbiota specialist, and also Michel Mistou from INRAE, Annick for BCCM, Marie-Z Grunwald at the Verstadik. I'm really sorry, I would say BCBS, it's much easier for me. I'm sorry. And also, we have also to thank people that help us for ISPI, like Theodora Abu and Francisco, who participated to the question and answer this morning. I have to thank Marta and Irae, Alessandra and Leslie at Alpha Visa, and also Luis. I know who he is. And so I hope it was really a super meeting.
Thank you so much to everybody.
So I think that so we have the pleasure to meet you all in person once soon, and I hope that you enjoyed this meeting. It was really a pleasure for me to be with you today. Thank you all.
Thanks to you.
Bye-bye. See you next time.
See you. Bye-bye.
Bye.
Allô, Alpha Visa? Merde. Ok.
Oui, je vous écoute.
Non, mais c'était pas prévu, mais en fait, merci. Et puis, je sais pas trop comment ça fonctionne après, ou je reviendrai.
Vous êtes toujours en direct, ne vous inquiétez pas, vous pouvez juste quitter la plateforme et puis nous, d'ici quelques minutes, on va terminer l'événement. Ok, très bien. Mais écoutez, je vous remercie et puis à bientôt. Au revoir. Et Je vous remercie.
Au revoir.
Au revoir.